we're going to ask Peter Gregory to stand up and give us a couple of minutes of introduction and some thoughts on the day, and then we'll get on with the program. So thanks, Peter.
Well, thank you very much, Tim, and it's a great pleasure to see so many familiar faces and for me to be back here in Dundee, particularly at the Botanic Gardens here, because shortly after I arrived in the Botanic Gardens in 2005, I was actually asked by the Vice Chancellor at the time, Alan Langlands, to conduct a review of the Botanic Gardens and as to whether it should continue to exist. So you know the conclusion of the review. It's a great pleasure to be asked to introduce this and to honour, really, all the work that Philip did through his career. The first time I met Philip was actually at the University of Manchester as his PhD Viva examiner. And I have to say, it came as a bit of a surprise to me to be asked to do the examination, because Philip had actually worked most of his time down at Leckham for it. He was registered at Manchester, but he'd done a lot of work with David Clarkson at Leckham. And it was only afterwards that I realised that the connection must be because I knew David Clarkson, and he thought I might be an appropriate person to challenge Philip. I remember the examination very clearly because it was just a great pleasure. The thesis was very clear. I think I'd examined probably about a dozen PhDs up to that point, but it was the clearest PhD thesis that I'd seen to that point. And basically at the beginning I said, well, look, we don't need to worry about whether this is going to be pass, fail or whatever. We can have a conversation about this topic. And I remember very clearly the excellent conversation that we had. Over the years past, I saw Philip, I think, at the various HRI reviews that were fashionable at that time. In fact, it was almost an annual event to have an external review of Horticultural Research International at that time, before DEFRA eventually owned up that they were going to pull the plug on the whole thing. But I didn't really talk with Philip very much at that time. I saw him, but we didn't really talk. But the second time I met him was an infamous day, the 5th of July in 2005, when various people tried to blow up people on the underground and buses in London. And he was at one meeting in the basement of the Department of Trade and Industry, and I was at another meeting. And our paths crossed, and I'd already moved up here to Dundee and just dropped a word in his ear to say, would you be interested? Would you be interested in coming up to Dundee? Because I wanted somebody to come here and head up the Environment and Climate Interactions Programme. And the rest, as they say, is history. So it's a great pleasure to be here. I'm looking forward to hearing more about Philip's life and Philip's work and talking with you during the breaks. So thanks very much for coming, particularly to those who have come from Slovakia and from the Czech Republic. And to all those of you who are listening online, goodness knows what time of the day it is. Thank you for being with us. Thank you.
First up is Dr. Paula Pongrad from the University of Ljubljana in Slovenia. And she's going to be telling us about some of the work she did with Philip when she was here, I guess, yes. On a Marie Curie fellowship.
Yes.
And so the title of the talk is Organ and Tissue-Specific Partitioning of Elements and Transcriptome Responses of Date Palms to Salinity. So over to Paula. Thank you.
Thank you, Tim. Thank you, everybody who has joined us online and everybody in the room. As Tim said, I'm Paula, and I did a PhD with Philip, and it was not on date palms. This was the work we did after my postdoc here in Dundee. And I need to give you some context before telling you about the date palm story. So I met Philip in 2012 in person at a meeting in Zurich, where I asked him if he would have me as a postdoc working with him. He was a bit reluctant at first, I must admit, but I was very persistent. So in September 2012, we applied for a Marie Curie Fellowship already, which was not funded at that time. But we repeated application in 2013, which was successful. And I came to Dundee 2015 and stayed for 2 years, good 2 years, during which time I shared an office with Philip, which was a very special experience because I was constantly challenged and had opportunity to grow really significantly. And I will be grateful to Philip forever for this opportunity. And during that time, I also took that picture, which Tim used for this event. He needed a new picture. There was a conference coming up, and he said, they want me to submit a picture which is not older than 2 years. And I said, let's do the photo shoot. So we went to the fields and we did some pictures. It was great time, unforgettable time when I met friends, not only colleagues, also friends. So we stay in touch all this time. And my family also enjoyed the time in Dundee. We did some outreach activities and my kids have fond memories of Dundee and also the bounty in the fields. So that was very good. During my postdoc, so I worked on Brassica oleracea, and the first year I spent bulking for seeds because we did a selection of specific genotypes. There were some seeds available, but I spent whole year bulking, and these were my plants. I was very proud because I've never done this before, but with help of several people in the group, I managed to acquire enough seeds to do one of my experiments, which were dealing with zinc and phosphorus interaction in root uptake. So I did several hydroponic experiments, but during bulking those seeds, I also did a soil experiment. So I wanted to use my time here efficiently. So Philip and I decided to do an experiment with red cabbage, a genotype of red cabbage, and we collected different soil types and grew the plants. And we showed that there was a very important, very significant effect of the soil type on the ionome or the elemental composition of those red cabbages. And this was our first publication with me as a first author with Philip. So I did hydroponic experiments on the subselection of Brassica oleracea. We grew them for only 2 weeks. We could show that they, again, as has been shown for a larger set of genotypes, that they do differ in zinc and phosphorus uptake. And also they were affected by different phosphorus supply in combination with different zinc supply. But we were looking also into what is the transcriptome responses in roots. And this was another paper we published together in a collaboration with Dr. Sina Fisher down in Nottingham, where I also did high throughput root phenotyping for those lines, genotypes. Of course, we did ionome. We looked at in greater detail in 2 genotypes. This was like one broccoli and one kale. These were our main candidates, our main genotypes we worked on. And of course, we could show also in hydroponics that based on all ionome fingerprint roots, which are painted here and shown, highlighted here in pink, their ionome differs significantly from the shoot ionome. We continued with RNA sequencing on these 2 cultivars. We were comparing different treatments for the 2 genotypes, and we could show that the kale genotype responded much significantly, had larger number of differently expressed genes, and we looked at gene ontology analysis and showed that there were certain groups of molecular pathways that are changing in response to phosphorus and zinc supply. These 2 genotypes were then also looked in for their root traits in response to their phosphorus in the phosphorus supply, and we could show that the kale has less secondary roots than broccoli. We did infrared phenotyping of the exudates and then did GCMS on those, and again show there is huge difference between these 2 genotypes. And we continued with also doing localization of phosphorus in roots, and again showing the differences in responses to phosphorus supply in these 2 genotypes. So this is shortly, and this has been published before, so there is a lot of there's data available for those that might be interested in greater details. But now I will move towards first outreach activities during my time in Dundee we did an art exhibition with an artist, Jean Duncan, and it was called The Beauty of Roots. And its first exhibition was at the University of Dundee, and we spent great time, a lot of time planning this, and were very pleased with the result. And then this exhibition was brought back to Slovenia. I returned to Slovenia in 2017. Sadly, the exhibition did not occur in 2020. You will all remember it was pandemic year, and nor Philip nor Jean could travel to Slovenia to help me prepare the exhibition. But then in 21, I had all the material shipped to Slovenia and made this exhibition. This was excellent opportunities to do some outreach also in collaboration between Scottish researchers, artists in Slovenia. So now finally coming to date palms. So when I arrived in 2015, I was given dates to eat in Philip's office, and he said, take them home and bring seeds back. And I said, okay, that's good, but why? And he was like, I'm joining a consortia on date palm studies, and we will be growing date palms in Dundee. This was not done by me, but by Gladys and Jackie, his technicians, and they were germinating seeds and preparing plants to be grown in hydroponics. But not only hydroponics, we also had salty pot experiments. No, I was just observer at that time. So these were 2-year-old seedlings purchased from date palm development and grown in Dundee glass houses and were exposed to salinity treatment. And initially it was 8 date palm varieties. And at the end of this experiment, roots were looked at. You can see huge root system. And they were separated into secondary roots and primary root. And of course, as you will know, elemental composition was determined. And here are some initial results. So this was not my work, but you will see why I'm showing this data. So this is for sodium concentration in the 8 different cultivars, and C is a control and plus Na is a salinity treatment. You can see really high concentration in secondary roots comparison to primary roots, and then a decrease in sodium concentration towards leaves. We similarly, of course, Philip did all ionome, looking at chlorine. The data here shows again that the secondary roots contain the highest concentration of chlorine, but the difference is much smaller between control and salinity treatments. So Philip then also looked at sodium translocation factor. And if I may draw your attention to the last variety, Sultana, you will see that the translocation factor, which is a quotient of sodium concentration in shoots towards roots, is fairly higher than for other varieties. And he was very interested in this variety. And the graph on the right-hand side shows that with blue symbols, that Sultana differs than the rest, even when we compare the plants that were pre-experiment. So the label P means purchase plants or pre-experiment plants. So he was looking into, he wanted to study in greater detail Sultana, especially he was interested where sodium is located, because so this plant is obviously able to accumulate larger amounts of sodium in the roots, in the shoots. So he was wondering where this sodium allocates on the tissue level. And this brings me to one of the chapters we published together, where we elaborated on distribution of heavy metals. This was a heavy metal paper. So we mapped or summarized for hyperaccumulating plants where those elements are allocated. And I was particularly interested in this because this is a technique that I use a lot in Slovenia. And Philip contacted me and said, let's do a localization study on sodium and chlorine in these date palms. And I grew plants in Slovenia. So this was in 2019. The two genotypes, the two varieties, Kalas and Sultana. So Sultana was the peculiar one, and Kalas was chosen because he's been extensively studied. So in these plants, they were also exposed to salinity or control treatment. And after 6 weeks of growth, I looked at root tips, root base, shoot base, and leaf base and leaf tips, and prepared cross sections of this after cryofixation, cryosection and freeze drying. So this is the approach we typically take to prepare samples, which are essentially cross sections, freeze dried cross sections of different parts of the plant. So this is an example on the right hand side of a cross section of a leaf of date palm. On the right hand side, this is a UV excitation used to excite autofluorescence in this leaf. So these cross sections are then brought to a facility at Josef Stefan Institute in Slovenia, where we have huge microscope. It's a proton microscope that enables determination of elements in a spatial manner. So this is a huge microscope, and the samples are then inserted in this chamber, vacuum chamber. The protons bombard the samples, and the detectors then can resolve which element is located at what concentration in certain pixel. So this is how the cross sections look like. You see on the left hand side, it's examples of leaves and roots, and they are inserted in this chamber. And after a few hours of measurement of each samples, we can then present distribution of elements, which I will show you in this slide. So on the left-hand side, it's Calas and Sultana. On the right-hand side, and sodium, chlorine, and potassium allocations were resolved. And the problem was that sodium concentration in leaf tips were really low, and also in the leaves were close to the limit of detection for the technique. But I will draw your attention to the root tip, where you can see large sodium accumulation in the cortex and not much of tissue-specific partitioning for this element in root tips. For example, I'm just drawing a colocalization of silicon in red and chlorine in green and phosphorus in blue for a cross section of the leaf, and you will see phytoliths in red here, located this here, hotspots of silicon and chlorine accumulation to fiber bands, which was a big surprise for us, and phosphorus location in the vasculature in the center part of the leaf. We continued to build on this type of experiment, and with the RNA sequencing on a third experiment we did, again on these two genotypes, and we did RNA sequencing, and you will see this was a big surprise. Very excited to see this. So on the left-hand side, I'm showing, so there are the two varieties, and Calas on the left-hand side, Sultana on the right-hand side, and brown here is root differentially expressed genes in roots. And you will see that in roots, Calas responds significantly. There is a lot of changes happening, but this is not the case for roots in Sultana. But by contrast, Sultana will respond more significantly in the shoot. And this is one of the reasons we think is that Sultana is able to have really good sodium mobility, but is responding significantly in the shoot, trying to sort these sodium toxicity problems happening in the shoot. So it's not able to stop sodium in the leaves, in the shoot, in the root. The image on the right-hand side, the graph shows now the comparison between the two varieties, and you can see that already in control treatment, which is shown here in red, there is large variation between the two varieties. So in both roots and in leaves. So they differ significantly also in already in control conditions. So it's really hard to point out certain genes that would be responding to salinity significantly. And this is one of my final slides. Almost it's a Venn diagrams showing first the salinity responsive genes in roots, comparing Calas and Sultana, and you will see there was only one common gene responding in both varieties to salinity in roots. There was more commonalities in leaves, but also when comparing control and salinity responses, so comparing Callas to Sultana, differentially expressed genes in roots, larger numbers were changed in both of the varieties. So this is a paper in preparation. Sadly, it's in the absence of Philip's insight, and it's been really hard to get this to this point. But since we've invested so much time and we think the story is interesting, we are trying to get this published. So this, the picture I'm showing now is the picture we took in another botanic garden. It was in Edinburgh, and Philip took me and all the colleagues. And this is Jean Duncan, the artist. We worked with the two technicians, so Gladys and Jackie, and Philip's PhD student at the time, Conrad, who will also tell you about his PhD today. And it was a really nice time together. It was a farewell, but of course, as you realize now, I've stayed in touch with Philip and also with others. And for me as a person, as a family, or in my family, this was time in Scotland was really amazing. And we come back regularly. And this is a picture from last year, staying in touch with both Gladys and Jean and other friends we met. So thank you very much for your attention. I was on time.
That's perfect. So, Laurie, can I check, are there any questions online first? No questions online. If you have any questions, please add them.
Raise their hand online.
So somebody's raised their hand. Is it Malcolm? Yeah, Malcolm, would you like to open your mic and ask a question?
Yeah, well, first of all, thank you for a beautiful seminar, which really illustrates the breadth of Philip's interests. And thank you also for the very personal accolade to him. It was very, very moving to hear. My question relates to your really interesting differences between the two varieties. And I was struck, I thought that the imaging you did with the sodium was really beautiful. And I was struck by one of the cross sections of the roots you showed close to the end. And it looked as if the variety which actually is able to to tolerate higher levels appear to exclude it in the outer tissues. So it suggested there were barriers which were in the mature parts of the root, which were keeping the sodium out. Would you like to comment about this?
Thank you for noticing this in this such a short time. And, you know, I spent hours looking at that, and I will revisit all the data again and look at this particular in greater detail. And in fact, there was lower concentration of sodium in the roots of Sultana compared to Callas. And it could be, yes, there was some hotspots of sodium seen in one of the roots, and thank you for pointing it out, but I will need to go back and so these were only representative cross sections, so I did repetitions for all, and I will need to check if this is something that occurs on all my repetitions. So if this is something that is really there and it's not just happening in this. Thank you.
Thanks, Malcolm. We've got time for a question in the room as well, if anyone's got any questions here.
Just,
Jack.
yeah, just thinking about the situations where we have problems with salinity. Another sort of real life biophysical thing is the water uptake and situations in parts of the world where you've got the salinity problems, maybe have irrigation. You've got a very kind of elemental nutrient sort of focus on the study, which is great. Can you put yourselves in Philip's position to answer the question about how we think about water relations in this context?
Yeah, because that's an impossible.
Yeah. Exactly. Thank you.
Yeah, do your best.
So in the experiment, as far as I know, we didn't measure anything apart from biomass and element composition in the experiment, or now the localization. So I cannot comment on this. What I can say is that those plants, there was no effect of salinity on their biomass. So I think they were not stressed in that sense, but I wouldn't know how this will affect the water uptake. Sorry.
Interactions are critical, right?
Yes.
Yeah.
Sorry.
Okay, we're on a
Thank you.
very tight schedule, so I need to move on to the next speaker. So thanks, Paula. We'll give you a hand again. And I think I mentioned at the start, it's a change to the program because Alexander Lutz is stuck in Amsterdam, or was. So hopefully he's on his way now. But Ali Carley is going to take over this slot. And I assume that we can see the slides online again. I'll just check, just in case. Can you see the slides online, Laurie? Yes, perfect. Okay, so over to Ali. You have 15 minutes or so before I start jumping up and down. So, and
make sure you do, because I might don't want to go over. Thank you very much for the opportunity to talk today. This is going to be very anecdotal. There's not going to be much science in here, see if you can spot it. But thanks for letting me give a bit of an overview of my time, my knowledge of Philip. And my interactions with Philip were quite intermittent in the early stage, but it turns out I actually knew him for quite a long time, which is probably why I'm here. He and I, well, actually the first time I could have met Philip, I didn't. We were both at University of Cambridge at the same time. He was a postdoc in Enid McRobbie's group in the Plant Sciences department, and I was an undergraduate student. But at least I don't remember, and I'm sure I would remember my first meeting with Philip if I'd met him there. It was Enid's lectures that really set me on a path and sparked my interest in plant biophysics and plant membrane transport. And that's part of the reason I then encountered Philip later on in my career. But yes, I managed to miss him that time. He was a postdoc looking at cation channel, transport through cation channels using the planar lipid bilayer method, which at that time was relative. It was he was one of a group of people who I think were quite pioneering in the application of these electrophysiological methodologies like lipid bilayers and patch clamping that are traditionally or more usually being used to study animal cell biology, were now being much more exploited for understanding plant membrane transport in plants. And so it's a really exciting time to be involved in that area of research. I was exposed to this methodology actually as a summer student after my undergraduate project. I'd already started moving down the path of looking at membrane transport or plant transport processes through my honours project, actually doing something very similar to your date palm work, so we'll have to chat about it afterwards. But I did a summer project learning this planar lipid bilayer method, and that sort of set me on a path to my PhD, which followed subsequently. But yeah, I didn't meet Philip at that time, and in fact, the first time I actually met Philip was at a conference. It was the International Workshop on Plant Membrane Biology, which was held in Germany in 1995 in Regensburg. This is before all our lives were digitally documented online, so I struggled to find any evidence online that this conference took place, apart from thanks to Rothamsted Repository. One of the colleagues of one of my PhD supervisors, Roger Lee, had an abstract which references the conference. So it did happen, and I shouldn't have been there. It was the summer before I started my PhD, and I was doing yet another summer project, this time at the University of York, in a group that was allied to my future PhD supervisors group. I was working with Julia Davies on fungal membrane transport, and I was planning to do a PhD with Roger and with Dale Sanders on looking at membrane transport in barley cells. But the summer before that, I was working with a fungus, and one of Dale's postdocs, put the idea into my head, well, maybe you could come along to this conference. We're all going, and it'd be a really great experience for you to go. So I went to Dale. I said, can I go to this conference? Will you pay for me to go? And I think he must have been so taken aback at the cheek of it, he didn't know what to say. And I ended up going. And it was a fantastic experience. It was I remember this meeting and other subsequent meetings, really kind of vibrant time in this area of research. Lots of lively discussions, some very robust debates, and it's definitely left a mark on me. Anyway, one of the social events for this conference was an evening cruise down the river Danube. There was a meal on a boat and some music and dancing, and it was a lovely evening. I remember coming onto the boat with a group of us from Dale's lab, and at some point somebody introduced me to Philip between me and the bar. And then we got talking about something. I think Philip probably challenged me about something, some aspect of my planned PhD research. I don't remember exactly, but we ended up having quite an intensive discussion. So you can imagine by the time I got to the bar, I was pretty thirsty. Anyway, it was an enjoyable evening, and we had other social events throughout the week. After that, my interactions with Philip were quite sporadic. Mainly through attending the same meetings, such as the annual Plant Transport Group meeting, which he convened for a number of years. After my PhD, I then transferred my interest in plant mineral nutrition to looking at how changes in plant nutritional physiology affect other organisms that interact with plants. And I spent a few years looking at how changes in phloem nutritional physiology can influence the population dynamics of insect pests such as aphids. I moved to what was then SCRI in 2004 with the aim of transferring this nutritional physiology approach to looking at insect-plant interactions to understanding how multi-trophic interactions occur in agroecosystems. And Philip arrived a couple of years later, as Peter said, to take on the role of program lead of the Environment Plant Interactions program, which was where I was based as well. Looking back, I realized that Philip and I had quite similar interests in plant nutrition, just coming at it from different perspectives. His focus was on improving the nutritional quality of plants for human consumption, particularly to address nutritional deficiencies. I was interested in how changes in plant nutrition might influence herbivorous insect pests, the ecology of insect pests and their management. Obviously, Philip is far more astute than me and spotted these similarities and interests, and he was very kind enough to invite me to co-author a couple of articles building on our shared knowledge of some of these mineral nutrients. Perhaps more importantly for the wider institute were the new capacities that Philip brought in, and as Peter said, inviting Philip to the institute to ask him to set up this area of research had huge benefits for lots of us. He brought in new methodologies and equipment for processing plant and tissue samples and importantly for analysing them for mineral nutrients, which we hadn't really been able to do before he came. And I was very interested in helping facilitate these methodologies, so I worked closely with Philip and with Jackie Thompson, who's his research assistant, to set up the equipment, the protocols, the calibrations, the standards, and so on. Just to give you an insight of and this methodology has really been the basis for lots of research from the Institute ever since. And just to give an insight from the impact that it had in terms of our research on pest ecology and management, I just want to give you one example. At the time, there was also a relatively new arrival, a colleague and collaborator, Scott Johnson, who's now moved on, but we worked closely together on several projects. And we were interested in understanding how, because plants are attacked by multiple different organisms, and often some of these, this attack occurs below ground where we can't see it. We were interested in understanding how insect pests might interact with each other indirectly through changes in the host plant physiology. And in particular, one example is this pest on the bottom left, right-hand side here, the vine weevil, the larvae of which attack the roots of a whole range of horticultural crops. It's a big pest globally. But it's quite difficult to work on to know when this pest is a problem, and sometimes we thought that it was interacting with some of the shoot pests that we could see above ground. There are a couple of different hypotheses around that have been devised to explain how below ground feeding by insect pests might affect shoot herbivores above ground, and I've outlined them here. The nutrient stress response hypothesis proposes that herbivory of plant roots leads to the plant becoming nutrient and water stressed, and that leads to senescence processes that lead to protein degradation, release of free amino acids. And that this release of nitrogen-rich compounds then benefits shoot herbivores because it improves their nitrogen nutrition. Alternatively, the defense induction hypothesis suggests that root damage by root herbivores will actually have a negative effect on shoot herbivores, because root herbivory sets off a train of defensive processes in the plant, which includes the release or synthesis of defensive chemicals that are toxic to shoot herbivores. While Scott was at the Institute, he and I carried out a number of experiments to test various aspects of these hypotheses on a number of different study systems, one of which was blackcurrant. This isn't actually blackcurrant, but it's the only picture I had. Blackcurrant being attacked below ground by vine weevil larvae and fed on the shoots by aphids, phloem-feeding aphids and chewing sawfly caterpillars. As we would have predicted, on the right-hand side, you can see that when blackcurrant plants are exposed to weevil larvae, the W treatments in the bottom, there is a significant reduction in root mass. The roots are damaged. And this correlated with an increase in the abundance of aphids on the weevil-treated plants. If you look on the right-hand side, the middle graph there, compared to no weevil controls, but a decrease in the relative growth rate of the larvae of sawflies. The graph on the figure on the right-hand side. When we look at the nutrients that are changing within the plant during these treatments, we found that leaf phosphorus concentrations were lower in the vine weevil treated plants. So on the graph on the left-hand side, the black dots show the vine weevil treated plants compared to the weevil-free controls, which are white. And this seems to correlate with the reduced larval growth rate of the sawfly larvae. And when we looked at the amino acids, those nitrogen compounds, particularly the essential amino acids, which, as the name suggests, are essential that the insects can't synthesize themselves, there was, on average, there's a lot of scatter there, but on average, there was a small increase in the proportion of essential amino acids in the leaves of vine weevil treated plants, the black dots, compared to the weevil-free controls, which are the white dots, which seem to correlate with the increase in aphid abundance. We also looked at the defensive chemicals, the phenolics are the main defensive chemicals in blackcurrant, but we didn't see any change in the leaves, a small change in the roots. There was no pattern that related to changes in herbivore performance. So overall, we summarized from this particular experiment, the predictions that or the observed effects we saw were a decrease in root mass with root herbivory from vine weevil larvae, an increase in a decrease in shoot phosphorus concentration, maybe an indicator that nutrient uptake was suppressed, and an increase in the proportion of essential amino acids in the leaves. And these are all effects that we would expect to see if the nutrient stress response hypothesis holds true, which is what we saw in terms of increased aphid abundance. However, we did see a decrease in sawfly larval mass, although no change in shoot defensive chemicals. So, and it's possible and it seems that from this research, but also a wider set of experiments carried out by us and others, that the way that shoot herbivores respond very much depends on the feeding guilds that they belong to. So finally, my talk promised a little bit more about aphids, though, because it also suggested that Philip somehow got involved with these insects. And this came about because Philip was very interested in some of his work in measuring mineral nutrients in phloem sap, and particularly in biofortified plants that he had biofortified with zinc. So we had a discussion about the pros and cons of different methods and ways you can sample phloem sap. Obviously, aphids are phloem sap feeders, so they can be one way of accessing the sap. And so that's what we did. We used aphids as live samplers of the phloem sap. You can collect their egesta, known as honeydew, either on an artificial substrate, or you can cage aphids onto leaves and collect the honeydew as they release it, and then to extract that honeydew and put it through ICP MS for analysis. So that's what we did. I think if I remember rightly, it was zinc biofortified tobacco that we tried this out on to begin with. But when we put these samples through the ICP MS, we did get some data, but unfortunately the levels were really right at the limit of detection and confounded by background. So we didn't get a nice data set from it, but it was fun trying. And finally, something I didn't allude to in my talk title, but something that Philip did that has actually had a huge impact on our research in ecological science department, as we're now known. Back in 2013, a colleague of ours at the Aberdeen site in the Plant Community Ecology Group, Rob Brooker, approached Philip asking, because he wanted to find ways of bringing together mutual interests of the upland ecologists and the crop scientists and crop physiologists at the two sites. So Philip and Rob kind of identified as intercropping or growing mixtures of plant species together as an area of potential mutual interest. And they worked to run a workshop back in 2013, which include people from the Institute, researchers from other Scottish research institutes, and through Philip's connections in China, he brought in collaborators from China, where of course intercropping research was really well established. The workshop was really successful. It triggered a lot of follow-on research and also a review paper that Philip and Rob were lead authors on, which I'm sure Philip would be delighted to know. I checked the other day, it's had more than 700 citations. And this has obviously led to a whole area of research, both in intercropping, in cover cropping, and related research. And this is just one example here of an EU project that we won funding for a few years ago. So I just want to end there and to really say that Philip's legacy for us as researchers at the Institute and more broadly are so broad. I think we've already picked up on that with the first few talks today, ranging from membrane biology, mineral transport, through to plant-insect interactions, through to mixtures of plant species, and so much more than that. So thank you very much for giving me the opportunity to give that accolade to Philip.
Thanks, Eve. Really great again. So have we got any questions online at all, Laurie? Can you see? No questions. If anyone would like to put their hand up in the online, then feel free to. Any questions in the room? Obviously, people need coffee. No.
Thanks for that. Your weevils, when they're grazing on the root, are they grazing? Are they taking whole roots out, or are they just grazing on the surfaces of the root?
That's a good question. I think they probably start with the finer roots, but honestly, if there's a heavy infestation, they will just go for it. You can literally pull a plant out and it's got virtually no root left on it. It really depends on the level of infestation.
Adam.
When you're using aphids to do that, is there a technique where you can sort of wax the body off?
Yes, yes, there is. Yeah, stylectomy. Yeah, we discussed that, and at that time we did have the equipment to do it. The quantities you get are tiny, phloem sap. With honeydew, because the aphids are alive, they are feeding, and they can actually produce prodigious quantities of honeydew.
What's wrong with the technique then? Why is it so variable?
So what's wrong with the?
Why is it so variable? You have huge variation.
Yeah, I mean, it was just really a pilot system. I think the problem was that we were using tobacco. Miser's persicute, peach potato aphid, does feed on tobacco, but it's a subspecies. Well, they don't probably call it that, but it is a particular ecotype that feeds well on tobacco, which we don't have in Scotland. So I think the problem was that it just wasn't feeding sufficiently well, that it was producing enough honeydew that we could measure, because obviously in some samples we were lucky enough that we got enough to measure. So I think it was more to do with the aphid than anything else.
Pete, you waved a little bit. You got a question?
I'm just curious about the fact you didn't find any differences in the boy, but I guess I could have disguised differences of types within your.
It could have, yeah. It was just a total assay. Yeah. That's true. And actually, well, as you saw from the sawfly responses, definitely there was a suppression in sawfly relative growth rate. Something will be underpinning that. And it's quite possible it's just the composition of defensive chemicals rather than the total quantity.
There seems to be a concentration of research of looking at phenolics and background. What extent
Yeah, I mean, we did look at phenolics below ground, and we did actually see in one of the treatments, I think it was weevil plus aphid treatments, we saw an increase in root phenolics concentration in the roots, which, if I remember rightly, didn't seem to relate with any changes in vine weevil performance. But yeah, their phenolics are definitely present in the roots, and they could be impacting below ground herbivores.
Well, we move on. Are there any hands online at all? No. Okay. So if we're ready with the slides. Yeah, nearly. Then we'll move on to the next speaker, who's Dr. Lionel Dupuy, who's joined us from all the way from Spain for this this event. So he was here at the Hutton for many years.
About 13 years.
13 years, but moved on to Pastures New maybe 2 or 3 years ago now.
Yeah.
Yeah. So to Nica and Icabasque in Bilbao in Spain, where I'm sure the weather's much better. So.
Temperatures, well, yes, but in terms of raining.
Okay. Over to Lionel. Thank you.
Right, thanks, Tim. I mean, it's really good to be able to share what it was like to work with Philip, and so I mean, my objective is to do the same, essentially explain a bit to you what it was like to share some of his many research interests. And the one that I'm going to talk about is models. So models as of mathematical models. So you try to formalize a process that in turn explain how the plant responds, how the plant grows to its environment. But also model system, which is this simple experiment that helps you decide and understand what these processes are, so that when you make the model, you can use first principle as opposed to being purely empirical. You try to use the first principle to build your understanding at a higher level. So just to put things into context, this work started in 2007. So that's how things look like. So you can see from the furniture and from the curtains, and so what was the style and what was fashionable at that time. So Tim, you were nice and handsome. I clearly had not a single white hair. That's interesting. So Peter Gregory was our director, and David Hopkins was the science director, and he was throwing presents at us in Christmas lunch. And yeah, obviously Philip was always there to this Christmas lunch. And at that time, so I was recruited to work on this sort of model, so functional structural plant modeling. So the idea is that you essentially set a rule as to how one organ or one meristem or something behaves and interact with others, and then you give that rules to a computer, so it's very computer intensive, and essentially you let the thing run and you observe what's happening at the end. So it's very much a numerical experiment. And obviously it was the time like computers were performing now, so you could do very fancy computer graphics, and it looked very real. So that's not what happened. And that's essentially because having lots of discussion in Philip's office, like I'm sure lots of you had, I think this sort of approach didn't quite represent what we were standing for in terms of science. And I think because personally, I think I shared this admiration for the older generation of scientists, with what they were able to do, the science that we're still using, and with the level of resources that they had, which was very limited with comparison to computers and super sensitive instruments and so on. So the old botanists, they were already knowing that, for example, the root cap was where all the senses were. So just dissecting, you remove the root cap and then nothing responds. They were already experienced with auxins. Like you could create a lateral root by scrapping a bit of auxin on the surface of the root. I mean, Liebig's law minimum, I'm from the 19th century. I'm sure everybody who worked a bit with Philip has. So how a simple law actually makes you understand very quickly what the plant needs in terms of nutrients. Drew's experiment in one paper, essentially, you would know how all the major, what sort of the response you could expect from a root when it's exposed to changing nutrients from the soil, from all the major microelements. physiology, so I know far less. I mean, Philip was always explaining me how the whole plant works by the concept of organs trading nutrients and things. So again, things that comes from ages ago and still is the base for you to understand how the plant works. And hyketenuse, I'm adding it because I don't think we discussed it with Philip, but that's one of the one that I liked a lot about root system. So when I started making the model, so in the end we went back into, okay, what was wrong with the other models? And you see that there were already quite a lot of very nice ideas. So hyketenuse that I'm talking about in the 70s, so no computer, nothing. So you look at the root system as a demography of root tips, right? And essentially with very simple relationship, you can know that, okay, my primary root grow at this rate, so I can calculate how much length I'm getting. It's producing this amount of lateral root, so I'm gonna have, and so on. And what you realize is that actually you get a very nice polynomial relationship that describes you how the root system grows, and you have all the different branching orders, all the root length and so on. A very simple formula. Yeah, and then the model came into bringing more spatial approach because the demographic model is not spatial. So, well, if the root grows, then it has to grow from where there's root to where there's no root. So they introduced things like diffusion equation, including a source term, which is the biomass being pumped into the soil. And then a lot of these sort of model went and be developed about how you adapt them to a real condition. And then in 2008 started to appear. So we had already started that, but it was nice to see that people keep thinking about improving this simpler model, but one that you really understand how things work. And this is a tip growth model. So people knew that you can, by this principle, look at the growth of root system if you focus on the root tips. So based on that, if you want to represent the root system in its entirety, like the FSPM were doing, but with equations, then you need to find a way to represent the topology, and that's what was missing. So essentially what you need is you need three types of density. So you need the root tip density because that's your engines, that's what produce matter. And that's what is being modeled. Everything else is calculated, the length density and the branching density. And that's the important part, is the branching density, because that describes your topography, how things are getting connected. And the other second important thing was to include the direction of growth, because otherwise you just have a diffusion, things that goes in all the direction, but that's not what happened. The roots, they have a direction, and based on the angle of the branching and the gravitropic and so on, you're going to explore different area of space. So then you do then what everybody has been doing for centuries, which is you write the flow and how things are conserved, what's getting, what's get out. That's eternal, it's not changing. And except that you use these sort of densities. And then what results from that is that you have a flux of meristem that propagates like a wave. It's not a diffusion, it's a wave. And what gets behind the wave of root tip is your architecture, the branching, the way it's branching, the length, and how it is being distributed, right? So that's for the theory. Now that's it, it's finished, no model anymore. So just to convince you that this is all what you need. So imagine these two different types of root systems. Well, if you have a dichotomous, if you have a herringbone type, so one primary root and lots of laterals, well, this is how your branching density on the left and your length density on the right are going to look like, okay? And the wave is this line that propagates at the oblique. So sorry if you can't see my hand here, but that's the wave that produces this sort of, and you can go for every sort of topology and you will have a wave, and behind the wave you will have a topology and a length distribution. And we went also into, well, what happens, for example, if you want to reproduce Drew experiments, or if we want to program different sort of interaction, and each time there's a way, because at the base of it you have the basic fundamental process of elongation and branching, okay, which is right. So in Philip's head, I think from the start the point of the model was, well, okay, to understand, to show that you understand, but he had also the big project of trying to understand the genetic variation in root system architecture. Root systems are very complicated, really bushy and things. So how do you get to know what changes from one genotype to other? So the idea was that you use the model to tell you what are the parameters that generated these differences. Okay. And the first thing that we did to try to prove that the model were good enough is say, can we use it to reverse engineer the root system that you observe from mini rhizotomes, so different tubes put in the soil, which you put a camera into it, so you capture a bit of root hair, a bit of root length growing around the tubes, and based on that, you just have a kind of an estimation. So we apply the model to this sort of data, and we would get very plausible parameters for root growth in terms of elongation. So I don't know if I can point out with but I mean, in the equation, what you can see is that this is a closed form. This is a formula that everybody can use. You don't need a complex software. You put this formula, you feed it on your root length data, dynamic root length data, and you get the elongation rate, the branching rate from it, from a very simple model. Okay, and you get some parameters that are what you would measure if you go and. So then we went at, okay, what do we do now to start to actually study the genotypic variation? So at that time, there was not that many of these fancy root phenotyping or high throughput phenotyping platform. There was maybe only Xavier Drey, who I have to admit was he himself built a robotic platform with lots of equipment, plants moving, and but I mean we didn't have anything near the resource that was needed to build this thing and the time and things. So we had a PhD student. So that's what we had. So we started to stack scanners in a glass house. So the scanners, so the plant were growing in a pouch. So on the top of a germination paper that was in the nutrient solution, so we could do nutrient treatments in the system. And the scanners, they have this thing that they produce very high resolution imaging. So we could see root hairs from our system. They're very easy to automate. You connect to a computer, so you acquire lots of data. So with one PhD student, we had a high throughput phenotyping facility where we're able to acquire data every hour. We didn't because it was created lots of data, but we could. So, and we could then started to generate lots of image data sets, and we actually had big image data sets. So largely brassicas and barley, but I think there was lots of other projects also that I was not necessarily involved. So we had quite a lot of data sets. Also not acquired all by scanners, but also from other ways. And this was the material to start saying, okay, how are we going to use this data to learn about genotypic variation in the root system architecture? So we're using, well, either already made software for tracing roots, for example, on the top right, but we're also investing into how can we save time, because this takes ages. So we started to develop our own also image analysis software, and how do we click quickly so that we can process all these data sets, and how we then find out what sort of error the system is doing, and in terms getting some of the traits like angles, length, and things like that. And then at the end, once you get this data set, so you've got lots of points and lots of branches and things. So that's when we started to use the model. And we developed this sort of algorithm where from all these image segmentation data set, we would first start to build the density. So how is the space occupied by the root with the different angles and so on. So the same representation that the model does. And then after, what you do is you start to use an optimization algorithm. So you give an initial set of parameters and say, is this good? Okay, yes, no. If I improve in that direction, does it improve, does it not? And so on. And you loop up to a point that you do not find anything better. And that's your root growth parameters. And at the end, that was the ultimate goal. But there's other step as well when you really start to do the genetics, but that's beyond me, and I'm sure some other people are going to present that. But we had some very, very nice data sets, and particularly the, I mean, Michael's data set where we could look at lots of different traits and extract primary root lengths, lateral root elongation rates, and which genotype has more of one or the other traits. Okay, and after that, I'm sure there was some mapping and some genetics done, but I think, as I said, I'm not getting that. Right. Okay. So what's the legacy of all this? So I mean, obviously, there's the science. There's what you have done. So I mean, I can say on my side that we are still using this sort of approach. So in terms of the model, we did like how the water flows in this sort of density structure. More recently, we've used it a lot. We're studying in water infiltration, so we've made models of water infiltration and how the root system favors the flow of water through the soil after a rainfall event. And again, we use the same representation of the root system because it's much simpler to run a simulation that running one root system than having a very computationally heavy simulations that takes time, but that you also have to repeat because you don't have a mean root system, you have one simulation. So, and we find some very nice results that shows that actually the root system might be important not only for how good it is at extracting at this particular moment the water, but how good it is at redistributing it. That's because that's a way to avoiding loss as well. If you have a root system that keeps all your water, that doesn't favor the water going down, then you're going to have more evaporation. Or the opposite, you might get the opposite situation where all the water get lost by deep percolation. So this is an interesting trait to consider. It's like, how does the root system favors keeping water around by orientating the flow? I mean, we've had with Philip, we've done quite a few other models as well. Some got published, some others, like root hairs, for example. Like, again, how do you represent what the problem is with the least minimum amount of parameters and calcium transport and so on. Now, I would really like to insist that the legacy is not so much into what you publish and what you've done, but it's in the people and the influence that you have on people. And with Philip, we had so many projects that was summer students, high school students, undergrad, master, so many different opportunities and of setting up science festival, interacting with different sort of societies, art exhibitions. So Paula was explaining everything, so I'm not very good at keeping all the pictures, but this is an example of some of these science festivals and one group of summer students that we're working on on the project. So thank you very much. So I'm just going to wrap up. So this is really just, if you just have to look at the page of Philip and to realize that this is nothing in terms of what he has done. But I think despite that, I think model was very important in Philip's mind, because I can cite the anecdotes of when I arrived. So Philip took me on tour. So he went to Warwick, went to Nottingham, I can't remember, to other places. And I remember once I was sitting at lunch or something, or in the pub or something, and I heard people talking. And they were saying like, oh yeah, Philip and his modelers, and they always keep them close. And so I found it. So it's just a little anecdote. So it didn't weigh much in terms of citations and publication, but it's something that I think helped him think also, and how things really work. And this, I think, is the impact is, again, is all the people that has been influenced by him, by the discussion, by the thinking. I think is more than the actual direct papers that gets up. So yeah, I'll just put a few papers that we drew. So thanks very much.
Really great again. So any questions online? If you have any questions online, please raise your hand. In the meantime, anyone in the room would like to pick up on any of the particular maths details in the models or anything that you wanted?
I might not even remember.
Peter.
So yeah, that was really good, Leonard. I had a particular question, which was you stressed the importance very early on of knowing the angle of the roots if you want to do distribution. And then later on, you showed a picture where, as we see all the time in botanical drawings, the angle generates the initial growth, and then the roots start to turn downwards.
Yeah.
So what are your insights now on what causes that to happen? When and what causes that to happen?
Are you talking about gravitropism or the switch that sometime might happen? Gravitropism, I mean, I can speak a bit about gravitropism. It's something that is very curious to me because so when a root is gravitropic and clearly horizontal, you get a very strong response. The key thing is that when you're not completely vertical and not completely horizontal either, then it seems to be a lot more less straight, the root doesn't seem to be that strongly inclined to go vertically. Yeah. So I don't know if it's exactly what you were talking about. So maybe I can find a.
It's always struck me as so when you look at the botanical drawings,
yeah,
the things continue growing, and at some point they start to become geotropic and turn downwards. But it's done. They don't all turn at exactly the same point.
Yeah.
Or time in terms of their growth. And I just wondered what insights you've got into how do you generate that in your model? I
think if you had some insight into developmentally what's happening, I think in this case it's more like that an embryo takes a bit of time to get into the full the complete way of working. Yeah. And I think there's a bit of a lag before there's going to be a response, and you see it in lots of different ways. So that would be my interpretation without knowing exactly what's happening. But if you look at the transect of the soil and where a plant is growing, is overall as a whole, you of the cereal in particular, you really see that there is a kind of tendency. If you look in detail in one root, you might not see it, but as a whole, you see that there is a kind of parabola. I'm going there without being completely vertical, but I have a trend there.
So hello everybody again. Welcome back. Hope you had a good coffee and a good chat. So this session will hopefully run for an hour. One of our speakers hasn't quite made it yet, but hopefully they might appear in the next 40 minutes. So to start the session again, I'm really grateful to Tim for doing both working session this morning and also doing a talk about his work with Philip.
Thanks, Tracy. So I believe people online can now see our faces as well, so apologies. So I'm going to give you a bit of a story about what I've learned from Philip about impact. So you might be expecting to talk about how roots are the most important interface on the planet. I'm not actually going to do that. It's just an example of the kind of way you can frame your science or frame your ideas in a way that's engaging to people. So this is something that I think I learned a lot from Philip in many hours spent in his office on a Friday afternoon, listening to how we need to be really impactful with our research. So, and I'm sure again, many of us have been there. So there were some things that really stuck in my mind that Philip said to me in those sessions in the office. And some of them are written out here, basically. So it's really important that we, as scientists, we do this, that we engage with everyone who's interested in our research, and that we generate impact in many ways. So Philip always used to say, give the reader something they want to cite. So it's really important that people want to cite what you've done. So I'll give you an example in a short while how we took some data to generate a figure that everyone would like to cite, even though it was just in an editorial. So he was really conscious that every single thing that we put out there should be impactful and should have generated some useful output, not just throwaway statements or so on. So give them something they want to cite, give them a statement, give them something, something that's a catchy statement for people to use, images that people want to use, for example, and summaries of information, so tables with useful information in them and conceptual models and so on. And some of the papers that have been talked about this morning have those elements in them. They have things that people really want to get a hook on them, and then they cite the paper, and it makes that work much more impactful. Anyone who worked with Philip would know that challenging the dogmas really important. I mean, that was basically what he did. He didn't want to reinforce untruths. It was always about finding those dogmas and breaking the dogmas. So take every opportunity to produce an impactful output. So, like we say, we worked very hard on producing editorials for special issues, even though most people would just make some throwaway statements at the start of those. We worked very hard on those because it was an opportunity to get a message across rather than just a throwaway piece of writing. And as everyone knows, Philip was very keen on making sure that the writing was very good. So, it doesn't have to be a Nature paper to be impactful. And I think anyone who's worked with Philip knows that was his mantra, I think. And it worked for him. So, I mean, most highly cited in his field for several years. It's a testament to it. I looked at Google Scholar the other day to see where his citations were at, because I knew you'd probably want to know. Currently in Google Scholar, it's 45,000 plus citations, and 3,500 this year alone. So, he had impact, and the way he thought about these things gave him the impact. So I'll just give you some examples of how we worked together, and I'm sure that everyone in this room and lots of people online will have similar stories. Work together to try and produce impactful things. And in fact, the author of this paper is stood at the back, Jack. So I thought I'd show you this slide. So this is an example of how you illustrate something, simplify a big concept, simplify something that is really complicated. And Jack was the first author on that, but I remember we sat in the new seminar room at Hutton for many hours trying to work out how to conceptualize the impact of lime on many different processes, from molecular scale all the way through to ecosystems. And we were scratching our head for hours and hours about this, and I think Jack scratched his head for much longer than anyone else on this. But in true Philip style, he sat at the back quiet for a bit, and then suddenly comes out with a really insightful way of putting it. And essentially, I think my memory of it is that this is Philip's concept of how to deal with it. So it brings in that element of time and space, and understanding how a simple thing like pH change in the soil can have a big impact on everything over a long time scale. So again, it's all about that ability to show something very complicated very simply, although it's not particularly simple. But a testament to that is, I mean, on these slides, I put the number of citations again, because I thought Philip would like to see them. But so that's how many times that particular paper has been cited. So it's generated a lot of impact. Similarly, myself and Philip, the International Society Root Research meeting that we held in Dundee in 2012 was a great success, and we're here today because of it, because we generated a profit from that conference, and we're able to run this Dundee Root Medal Lecture every year because of that profit. Another output of that particular meeting was a special issue in Annals of Botany, and we produced an editorial. And again, like I said, it was an opportunity to find some interesting data to present and really generate some impact, and that's what we did. And myself and Philip sat in his office Friday evening, Friday afternoon, after Friday afternoon, after Friday afternoon, looking at the FAO records on consumption of different foodstuffs and their sources and the mineral nutrition of humans and all these kind of things, just to demonstrate, because we have this very strong idea that plants were the most important thing. So plants, and therefore roots were the interface where all of these elements were moving through. And we wanted to demonstrate that plants were much more important than anything else, than animals or any other sources of food. And essentially what we can see is that with that painstaking look at all that data from the FAO records, we were able to construct these figures, which are useful, and people cite them still. So you can see here that that total intake of calories, dietary energy, protein are all mostly impacted by plant products, from terrestrial plant products rather than aquatic products or terrestrial animal products. So the green bars represent that. Similarly, for the whole range of mineral elements or essential elements for humans, again, they all come from plant products rather than from animals. Similarly, also for the vitamins and the vitamins that are required, apart from in the case of vitamin B12 and vitamin A, where animal products provide those things much more readily. But this was basically just a clarion call for plants, to say that plants were really important to us as a species, and therefore we needed to understand how they took up these elements and utilize them through the roots more readily. And this, like I say, was for an editorial for a special issue. We spent hours on it, and it was really, really great. Similarly, for this special issue, we looked at the global data sets to look at the how different elements move through the wider environment, again to demonstrate that plants were really important as a catalyst for a lot of these dynamics. And we looked at carbon, we looked at phosphorus, we looked at nitrogen, we looked at sulfur, and we wanted to be able to contrast how different elements were more important, or plants were more important for different elements, or for the fluxes of different elements within the environment, but also just to really demonstrate that plants and ecosystems based on plants were much more important than, say, industrial and domestic causes of these fluxes, and to demonstrate that by understanding how plants work in their environment, we are able to affect these global dynamics much more effectively. So you can see that the size of the green arrows represents how important the plants are compared to some of the other parts of the wider environment. And again, for carbon and phosphorus and nitrogen, plants are clearly very important in those fluxes, whereas maybe for sulfur, we've got more of an influence of the sea and the soil. But generally, that interaction between plants and the environment is critical for these big global fluxes, and we wanted to make that clear and give somebody something to cite, essentially, in this area. Similarly, one of Philip's favourite ways of demonstrating importance in a particular, or impact in the impactfulness of a particular theory, was to use phylogeny to demonstrate either the uniqueness of something or the ubiquity of the idea. So in this case, we were looking at rhiza sheath. So the amount of soil that's attached to the roots of soil, to the roots of the plants, and the dogma was that only the poales, only the grasses have rhiza sheaths, whereas actually we thought that wasn't the case. So we did a phylogenetic study on this and demonstrated that actually, rhiza sheath is present in pretty much all of the different phylogenetic groups, and the poales aren't anything particular special, but the difference between the non-commelinid and commelinid monocots was stark, in that one set didn't have a rhiza sheath, didn't have root hairs, and the other did. So it's just using this kind of, I suppose you could call it blitzkrieg approach to demonstrating that something is either a fact or it isn't. Similarly, for there's some of us in this room, I think a few of us, we used to spend Friday afternoons in the living field doing a meta-analysis of root phenotypes. I remember we did it for months and months and months, and we pulled together all the data that we could find on the responses of roots to different elements, all the 14 different elements you find in soils. And we pulled together this matrix of information about how plant roots responded to different toxicity and deficiencies of elements and so on. And we drew this phylogeny up, which showed that plants responded in similar ways to different groups of elements. So, for example, in group 1, the plants responded in a similar way to potassium, boron, nitrogen, and sulfur. And they responded slightly differently to phosphorus. And then in a separate group of phylogenetic, or a separate group of physiological responses, iron, manganese, micronutrients, and so on, they responded differently to those compared to calcium, magnesium, and chloride. And I think this is a very powerful way of demonstrating that that plants behave in a similar way to certain stimuli, but differently to others. So it's a great way of demonstrating that, and it summarizes very easily a lot of information that took us a lot of time to generate. So similarly, conceptualizing ideas of what, say, for example, in this case, a root idiotype. So what you want in a root system for phosphorus acquisition, or what you want for potassium or nitrogen. And these different ways of presenting the information allows people to think about what it is that we need to breed for example, if we want to improve phosphorus use efficiency or nitrogen use efficiency, for example. And it gives people something to work towards. So I think Philip was great at really conceptualizing ideas and presenting them in the way that people could follow and people could grab a hold of and do some work and set some experimental hypotheses around. Similarly, Ali mentioned this paper earlier. I've got many more citations because I looked at Google Scholar rather than Web of Science. But this was the paper that Ali mentioned earlier, where we were looking at intercropping. And again, it was one of those situations where there was 20 people in a room, all talking about various things and all at odds with each other. Philip was sat there in the back, very quiet, and suddenly said, well, actually, it should be like this. And again, it's one of those, in my memory, it's one of those things where he basically stood up and drew the diagram that we needed to do. And everyone goes, oh yeah, that's right. That's how it should be. So, but again, like Ali said earlier, this has been a very impactful paper and lots of people have used it. And it's kind of driven a whole area of research, which is, funded massively through the European Union right now. So again, a great thanks to Philip for conceptualizing some of these ideas. And again, a similar example of something very visual that we produced to sort of describe what might be some breeding targets for root systems in barley. And some of these have been taken on, some of them not so much, but we certainly, that trade-off between root hairs and mycorrhizae is something that we work a lot on still. And just allowing us to conceptualize these ideas really allowed us to set the hypotheses to do the experiments, and hopefully other people have used it in this way as well. So that idea that you make a very complicated concept available to people to use and to utilize. And similarly, we spent long hours sort of working out how to provide a structure for research efforts. And this is a pipeline of research that we came up with specifically to drive research into understanding the response of plants to abiotic stress caused by climate change, with a view to driving innovation and breeding in this area. And this was particularly focused on potatoes, hence why we don't have so many citations. So apologies for that. But yeah, I mean, Philip was great at seeing the big picture and really crystallizing those ideas into something that's palatable and digestible by people. And I think that's a really great lesson for all of us to really look at our science and say, how can we communicate this in a way that people understand and people want to get behind? Because at the end of the day, you've got to convince people you're right to allow them to influence the way that they think. So yeah, Philip's lessons to me on producing impact are: conceptualize things in a novel way, smash the dogma, provide easily understandable insight, generate graphics and tables that people want to cite, and drive the research agenda by being the first to state the direction and the concept rather than following everyone else. So that's all I was going to say. So thanks.
So I don't believe we have any questions online. So has anybody got any questions in the room? I shall ask one.
Okay, right. I'm not getting away with it.
Other than the ones that you've selected, is there any specific ones that really changed the direction of your thought of the work that you were doing?
Yeah, I think many of the examples there did for sure. And I mean, I think there's probably many examples there that I haven't picked up on because they will have influenced people in the room in many different ways as well. So I think there's probably tens, if not hundreds, of examples of things that are in Philip's papers that lead people in the direction of the research that they want to do. And it certainly influenced the way we've done things.
Yeah, just a comment really. Just thinking back to that session that Tim alluded to, started to talk, and if you remember these words by Philip, that all this discussion and Philip standing up there drawing, or maybe before he started drawing, he said, "Look, it's a stone in the pond." I just remember those words. I think it's stone in the pond. What's he on about? And then he started drawing and explaining and just that.
Just clarity of thought. It's clarity of thought.
Yeah, we were kind of all over the place. You said, "No, stay in the pond."
Yeah, absolutely. And there's many examples of that in my experience, and everyone else's, I'm sure.
Well, I'd just like to say it's been really good to have you talk about 30 years ago.
Yes.
How do you like it?
Some of us are still doing it.
Yeah, it's a good lesson to perhaps to re-look back at a lot of the different papers that collated a lot of different information.
But for those who were in the living field, at the time it seemed like torture to be looking at all these papers and writing down all these different interactions. But in the end, it crystallized into something that's really useful. So yeah, get through the pain, basically.
Thanks very much, Tim. So next, I'd like to introduce Konrad Nuremberg from Humboldt University. Konrad previously worked in Dundee as a PhD student for Philip, and it's already previously been mentioned. So you get on.
Okay, so hi everyone, also online. In this talk, I will mainly focus on the PhD project that I did in Dundee, which was mainly under the supervision of Philip, of course others as well. Some of them are in the room or online, I've seen some names. And our project was entitled The Evolution of the Angiosperm Ionome with reference to the Caryophyllales. And I first start with some general background and introduction to the topic. Before talking about the evolution of plants and chemical composition, I want to take 2 steps back. So I first met Philip a long time ago at a plant nutrition conference in Germany, in Bonn. And it's been mentioned just now by Tim, actually, how he used to conceptualize things in a very, very intuitive and easy way. And at this conference, at that point, I was an undergrad student. I didn't actually know what the ionome was. I never thought about evolution. I was more interested in signaling and abiotic stress. And of course, that's one of the areas that Philip worked on and was very well known for. And I just said hi, didn't expect much. But he actually spent more than an hour starting with a sketch like this on a piece of paper, explaining plant science in general to me. And he combined all these areas, for example, root phenotyping, how plants can take up nutrients, how they are translocated within the plant. And I do remember, I think it actually was, this is meant to be an Arabidopsis, by the way, that he drew up. And I was quite overwhelmed, frankly, and a bit intimidated, because he combined all those areas spontaneously, just standing there, that I'd never even thought about. And I thought I was very interested in plant science. So that's something I really remembered, and which has come up now, that he was really good at that. But also, I do think it's quite remarkable that the highly cited researcher he was, which I didn't really realize at the time, he didn't mind taking time aside, even at a conference, talking for an hour or more to some undergrad students that he had never met before. Just enjoyed, or maybe enjoyed challenging people's intellect, but I think he also really enjoyed transmitting science to other people. I'm trying to go forward. So some years later, I was lucky enough to start my PhD with Philip, and to understand the evolution of the elemental composition of flowering plants, one has to like try and understand that flowering plants themselves are really young. So what you see on the left-hand side is a sort of timeline, and those stars, they mark important steps on the way to the evolution of flowering plants. And flowering plants themselves have only emerged about 200 million years ago and started diversifying even more recently, about 100 million years ago. And before that, lots of things have happened. So lots of the basic biochemical traits, or for example, the evolution of cell wall structures had already started before the first flowering plant emerged. And that's something that is important when talking about some of the results that were shown a bit, just to remember that actually a lot of stuff has happened before flowering plants ever came up. Nevertheless, today flowering plants are the most successful and diverse group of plants. There are many species, more than 300,000, and they can be divided into 64 orders, although this changes every once in a while. And it's important to remember that all flowering plants are assumed to have one common ancestor. So all the diversity we study, like in different species and even in genotypes within a species, they appeared after the emergence of flowering plants about 200 million years ago. The particular group of plants that we were interested in for this PhD project are called the Caryophyllales. And Caryophyllales is a very large order of dicot plants that sits right there, where you can see the red arrow. And they contain more than 11,000 species, which is equivalent to almost 4% of all flowering plants. So that alone made them interesting. But what's more is Caryophyllales is a very diverse group of plants. Some examples include many carnivorous plants like Venus flytrap, but also crops, especially in the Amarantaceae family, such as beets, like sugar beet, Swiss chard, they're all Caryophyllales, or spinach and quinoa would also be examples of crops within this order of flowering plants. But also they contain many plants with many members that are adapted to very dry and saline environments. For example, the Azoceae, they're also known as the ice plant family, or cacti. All the cacti are Caryophyllales. And well, it was surprising to me anyway, and I think it's to many, that cacti are on this level quite closely related to sugar beet, which is something many people don't expect. And it's important to note that an ancestor of the Caryophyllales is believed to have evolved in very dry and saline environments, which gives them which, because of the history, this might have triggered different adaptations, even in crops that don't grow in deserts these days. So again, trying to remember the history. So what did we actually do for this project? Well, since we were interested in phylogenetic differences between different taxa, we tried to minimize genotype by environment interaction. And this type of experimental setup was used. It's hydroponics, and it started many years before I joined the project. So in those large screens, many flowering plant species were grown in a nutrient replete kind of standard solution that was non-saline. And of course, not all species will grow in hydroponics, but it did actually work for some cacti, so it was impressive enough. And this allowed us to, well, and then we harvested shoot material during the vegetative growth stage. And this allowed us to compile this very large data set here that you can see, in which the shoot ionomes of more than 300 species were sampled and combined from 6 individual, like really large experiments. And for the first time, more than half of all the flowering plant orders were represented. And with a data set like this, well, you could basically do a lot. You see on the left-hand side the phylogeny of the orders that were represented. And what did spring out to us was that Caryophyllales, this order that we're interested in, many of them are adapted to very dry saline environments, as I said, have extremely large sodium concentrations when compared to all other flowering plant orders, on average. That's a bit of a generalization. But they also, for example, accumulated lots of other mineral elements, more than we would have expected. So they accumulated quite a lot of magnesium, especially when compared to like closely related orders that you see around it. So orange and red are high concentrations, yellow is not so high. And of course, verification was always a question that we were asked: how can we trust any of this? School hydroponics. But we did observe things that were well studied. For example, large sulfur concentrations in brassicalis. There's lots of literature on like glucosinolate metabolism. And I will now go on and highlight just a few of the key findings that we did draw from this combined data set. So first we looked at the sodium because it was the most striking feature. And actually this manuscript was developed in the first months of my PhD, when I hadn't even started my own experiments. But as was mentioned, Philip was always very keen to get new ideas out there once they were developed. So didn't want to hold back there. What we saw was that not all Caryophyllales accumulate large sodium. It's only, you see on the left-hand side, a histogram where you have at the bottom the sodium concentration in shoots on the log scale. So what you see, this cluster on the right, those species accumulate so much sodium in their shoots that most other flowering plants would have died already and never accumulated much. And the big cluster on the left-hand side, those would be Caryophyllales that accumulate more of an average sodium concentration that you would expect from an average flowering plant, which is a concept that Philip kind of introduced and enforced a bit. There's using this big data set that I just showed you of that heat map to try and define what is like a normal average flowering plant. Even if this plant doesn't exist, it then helps to compare different taxa to it. So we defined this trait first. What is abnormally large sodium in shoots for Caryophyllales in non-saline environments, right? So they accumulate much more sodium than there was given, and mapped it to a phylogeny in a very simple ad hoc way. We didn't use maximum likelihood or anything. We just counted. For example, Asoci, they're highlighted in red. It's the ice plant family, highly adapted to certain environments. Most of those species in that family that were surveyed in the initial experiments did exhibit the trait. We did this for various families and subfamilies. We could show that most likely this trait has evolved multiple times within the Caryophyllales, and it wasn't present in all Caryophyllales families. And so this idea was published really quickly. And we did, eventually, my experiment did catch up with the ideas that were already developed, actually. And we went back and did some redefining and some gap filling. So in the new experiment, we also, for the first time, did harvest shoots, and we grew more Caryophyllales species. So here colors will be different taxa. And what we did see is, on the top graph, you see those Caryophyllales that accumulate abnormally large sodium concentrations in their shoots. They also have a strong correlation between shoot and root sodium concentrations. And this red diagonal line is a 1-to-1 line. So all those that accumulate or hyperaccumulate sodium also have shoot to root quotients of more than unity. Whereas on the bottom, you can see lots of Caryophyllales that are more like an average plant. And they exhibit a classical like excluder sort of phenotype. Even when they take up sodium into their roots, there's this red diagonal line again where you see they all have shoot to root quotients of less than 1, so they don't really readily translocate it into the shoots. And this hadn't been shown and demonstrated before for sodium. So for the first time, we could actually define this trait similar to how it was done, for example, for heavy metals in the past. And then we looked in more detail at the phylogenetic resolution, everything. And we found, for example, that Amarantaceae, not all Amarantaceae exhibit this trait, but it seems to be only those that were previously considered a specific own family Chenopodacea. So beets, for example, even different beet phenotypes, they will all have this trait, but not Amarantaceae sensu stricto. Going back to this concept of the standard functional ion norm, we saw also that Caryophyllales had lots of magnesium when compared to other phenotypes or other orders that are closely related. And when we looked at it in more detail, that led to a completely different paper. That was really, the thoughts were developed by Philip. So when we look at the observed stoichiometries between calcium and magnesium, which had been done before, it's known that calcium and magnesium are correlated often in flowering plants. And it had been shown that different groups might exist. But he was, maybe a bit annoyed or curious because when talking to scientists from our field, everyone just sees their own area they work on. So when you talk to people, when he would talk to people who work on calcium signaling, anything to do with calcium immediately had to be about signaling. And people who think about magnesium, plant scientists, would think about chlorophyll. Right? So we know it's important for chlorophyll. So if you see a variation in magnesium, we assume it has to be chlorophyll. And he tried to break this idea a bit. And in this cited paper down there, he developed the idea that by really going through literature and unrolling everything slowly and logically, that lots of the calcium magnesium stoichiometry and variation could be explained alone by cell wall properties. And for example, on the left-hand side, we see those green and those blue dots. Those are commellinid and non-commellinid monocots. And there was this shift from type 1 to type 2 cell walls. And if you take apart those differences and their affinity for calcium and magnesium, if not everything, but a lot of the variation can be explained. On the right-hand side, we see dicots. And again, the gradient itself could be explained by using actually just cell wall chemistry alone. But what couldn't be explained was the orange dots. Those are the Caryophyllales, which I did mention also accumulate lots of magnesium. And in this paper, we hypothesized that this extra magnesium is in vacuoles of Caryophyllales. And this paper is basically an accumulation of hypotheses and thoughts that he purely built from literature by observing some data. And he suggests for each single hypothesis how other people should go out there if they want to and test them explicitly. So trying to shape the direction that research will go. I still have some time, okay. So one more idea that we tried to bring out repeatedly during this project was that the chemical composition of plants can also be used to build taxonomies. Taxonomies could be based on phenotypic traits and not just on marker genes. And we showed this on different taxonomic levels and for different organs. But here's an example of orders where we had at the top, you see a heat map and a clustering of the represented orders of 14 Brazilian forest species. And we took this and compared it to the same orders from our own big combined data set, but they were grown under different conditions. There were actually different species representing those orders. And we actually measured a different subset of the ionome as well. And yet the clustering of orders was so surprisingly similar that we thought, well, so maybe orders can be separated even across different environments. We combined this with large literature studies that you see at the bottom where we took plants or data of plants that were sampled from the natural environments. And again, could show that even when sampled across big gradients, geographical gradients, orders could be differentiated based on the ionome. So the ionomes of different flowering plant taxa are distinct. And of course, as often, even though I thought those ideas were new and hopefully even influenced by me, Philip could show that no, none of this was really that new. And he would often bring up papers of his own other people and he could say, actually, the idea had been demonstrated before, but people didn't realize. So here's an example of one of the Rothamsted long-term experiments. It's the parkgrass experiment, where different plots had been fertilized differently, I believe, since the 1850s. And he sampled species there, analyzed the ionome, and he could show that despite this environmental, like almost artificially strong mixing up of growth conditions, some families are very distinct. I can't really point easily with my mouth. So you can see, for example, the Poaceae and Polygonaceae separate from all these other families. And of course, some are more similar than others. Not everything is super separate. But those are the kind of ideas that you would keep coming back to. And in conclusion, for this PhD project under Philip's lead, we could show that ionomes of different flowering plant taxa on different levels are different and they are specific. And we could repeatedly show that the ionomes or the taxonomies based on ionomes are not correlated with existing molecular phylogenies. And this is because aspects of the ionome have evolved independently and repeatedly in different taxa. So in other words, it's a strong sign for convergent evolution, because if you remember this timeline from the start, there's a very complex prehistory of every species that we study now. And to conclude this talk, I thought about how it's influencing the work that I'm doing now after my PhD. The answers were very strongly. So the whole time I thought, even though it was a really interesting PhD project, it was very abstract. And I just helped out in some other experiments that Philip was involved with. For example, here some biofortification or fertilizer experiments for crops that he worked on. And of course, he worked on root phenotyping, as you have seen. And I think it's always fun maybe to do things that hadn't been done for myself anyway in a long time. So what I'm hoping to do is to shift back more to an applied ionome research and combine this with some of the fields that Philip has also worked on, like root phenotyping. I don't know how far I can take it. But yeah, with that, I would like to thank you very much and please ask questions.
Thank you very much, Conrad. Have you got any questions on that? Questions from the room? Lina.
I'm just curious about how difficult it is to work with so different for an experiment with that. What was the challenge? Just for us, nothing.
Standardization, I think, was an issue. So we know that chemical properties of a plant change depending on growth stage, many things, right? So even though we found an environment that allowed us to grow surprisingly many species, like Australian desert bush growing in hydroponics in Scotland, question was when to harvest plants. It was a big one, because we can't even count leaves if we're talking about cacti. So difficult compromises had to be made, and there were many debates and many sessions of Philip drawing sketches on paper. That was a challenge. And one other challenge was just the logistics of getting it started. So we tried to pre-select what species you might grow from which family, of which order. You maybe try to get some pro rata sampling, which can never be achieved, or there's lots of impossible compromises to make. And then you spend time, and then you order all those seeds, and you spend half a year preparing this experiment. It runs, and even like a year after the experiment was finished, some seeds would still be delivered little by little because they're very exotic. So that was just very practical issues. But it's important because people, they do often criticize it and say, well, it's not a fully balanced design, but it's impossible when you try to study all the flowering plants. You can't do it. Can do the best you can.
Just getting the right part of the plants to compare is very difficult. Leaf versus a cacti.
Well, yeah, we went for the shoot. I call it the shoot.
Yeah.
So everything, there were young plants, vegetative growth stage. We didn't wait until flowering, and we harvested what we called usually the shoots. But for example, in one paper, we called it then the leaf ionome, because it depends on one's perspective. A reviewer said for them, even if we grew tree species in there, what we harvest isn't really the shoot of a tree, because people think about trees as being something else. So for them, everything was leafy. So they asked us to call everything the leaf ionome rather than the shoot ionome. But it's, yeah, different people have different backgrounds.
Thank you very much. So just for the people online, Alex had some travel difficulties, but we're glad he's arrived now. So we had to swap Ali Carly and Alex's talk over. So
we are here to remember Philip, and we would like to start with something which is going back to history. And we are now here as an International Society of Root Research, but actually the root research special symposia started in our country much earlier. It was in 70s. At that time, I was only a student, young guy. But as we can see here, these were very long distances, time distances between individual symposia depending on the possibilities. But we do not know exactly when Philip joined these meetings, but we are sure that he joined in 1998 for sure, because we have some pictures from the history and we can see some of the participants and remember some of them, and maybe you know some of these people, maybe let's say Liam Dolan on the right side in the top and so on. And then it continued like this. Yeah, he was very enthusiastic giving some lectures, of course. And then it continued after some discussion during one of these meetings that he and Liam Dolan offered possibility to come to our university and to start giving lectures for our students, special lectures. And it continued a long time because for sure it was more than 20 years when Philip was visiting our institution, and he became visiting professor of our university in Bratislava. So we always expected him and spent several days, and the topics were changing during this time, of course. So one of his favorite, as it was mentioned also during the previous lecture, was phylogenetic approach and phylogeny and the relationship between the different species. So this is from 2016. And in the same time, or around this time, he was giving lectures also in neighboring countries, because he was in Austria, and then he was also in Brno in Czech Republic, where he was invited. And the reason why we combined this trip was that Philip wanted to see very much one museum in Brno. And so it was visit of the Masaryk University in Brno in Czech Republic in 2015. And then we went together with a group of our friends and students and head of the department, plant physiology, to visit this museum. So here we are watching Philip standing by Gregor Mendel. And if you go to this place, maybe I can recommend you to visit the museum, because you will know something more about the background of the genetics, we can say. And it's relatively easy to find, and Brno is close to Bratislava, so once you are in this region, nothing is so far. And then there was another, not the reason, but opportunity where we met, because one of our colleagues from Switzerland organized a special cost meeting, Jean-Paul Schwitzgebel. He was very much interested in toxic metals and the possibility how they come and go and are uptaken by plants, and how it goes later, and if it can be somehow removed. And it was not only about metals or elements, but it was also about organic pollutants. And this cost action was really very successful and joined very many people. And during one of these meetings, we met not only Philip, but also Martin Broadley, and then we joined for a publication. It is one of the really old excursions organized during these visits of Philip in our republic, in our Slovak Republic. And we visited several places which are quite interesting also to see the plants and the hyperaccumulators and so on. But then we were somehow invited, or we were allowed to join this famous publication about zinc in plants. You probably know about it. And the reason was that during one of these cost meetings, we were presenting structural characteristics of one of these hypoaccumulators living in our country. And we discovered what was described in this publication actually as a periendodermal layer with a very specific wall structure and composition of the walls surrounding endodermis. And I must say that it is still not completely clear how it works and what is the role of this layer surrounding apoplasmic barrier of the common roots for transfer of ions of several elements and or maybe to keep something inside of the steel and in the vascular system and so on. So we continued in this later, and we were very much interested actually in the role of endodermis as a barrier for the uptake or regulation of uptake and transfer of ions. And then we developed such a study that we have very many checkpoints, not only in the root, but it goes up to the shoot and to the flowers and seeds. And we must be happy because of this, actually, because not all is selected in the root, but also the transfer from the root to the shoot makes a very big difference in concentration of several ions. And we know that many of these toxic elements or toxic concentrations remain in the root. And then the transfer to the shoot is maybe only 10%. And another of these checkpoints is between the sporophyte and gametophyte, the gametophyte, and embryo junction, where there is another selection, what is transferred from the mother plant to the baby. And it is many times again 10% what is in the shoot. So actually we have maybe 1% only in embryo, and sometimes also in endosperm, but it depends. The surrounding organs which are protecting flower and the seed or the grain, which is actually not the seed, is many times full of these elements. But the proper of the embryo and sometimes also this storage tissues is a different story. So we must be really lucky because we know about the situation, how many of these plants are grown and in which places. And many times it is really the toxicity of the soils, or maybe when we speak about rice, is alarming. Fortunately, it is not all in our food. So that was some kind of continuation of this. We made such a scheme where it is in the special publication, and when we go to the root, so we can start from the root cap and look at the root hairs. They have also some kind of decisive function. One of the topics which was suggested by Philip to us was to look at the number of root hairs and the relationship with uptake of some elements, not only toxic, but also useful as phosphorus, for instance. And if I remember well, there was here a collection of different cultivars with different number of root hairs, and there was some relationship found, but we couldn't get the seeds because we started late and it was somehow abandoned. So then we moved to this, and we were developing this story from this cost action. And we have started actually with Maria Gregor. She was in one of these old pictures from 2020, for 20 2001, yes, where we have shown that she collected different, not cultivars, but let us say local forms of willows, and defined the uptake, translocation, and tolerance. And when we focused only on translocation from the root to the shoot, then we have found a very clear difference between what is on the left side, which are low translocators, and on the right side, which are high translocators. So then we continued, and Philip joined us, because then we wanted to see if such a function of the apoplasmic barrier can be active within the same root. So we exposed the root from one side to high cadmium concentration and zero from the other. And we were very lucky to see these pictures and then to put the publication together, and even more that it was on the front page of Journal of Experimental Botany. And here we are as co-authors of this in front of our faculty. So the story about root hairs actually continued with Arabidopsis. I myself must say that I'm not very keen in studying Arabidopsis. I prefer some other species. I do not want to call them normal species, but okay. But it was available and it showed nice results. So really, we were happy to have this publication. And then there was another aspect which influenced me personally very much, and it was publication by Professor Hodgson and his friends, and Philip White and Martin Broadley were involved, of course. And it was the variability in silica uptake in different species and the concentration. It is, well, it was, as you can see, this is a very old publication actually, but then I stayed in Japan and had an opportunity to study rice, which is very high accumulator of silica. So then it was easier for me to study some of these aspects and also the protective role of silica impregnation to the walls in various conditions. And one of the last impacts of Philip was when he stayed in my house and he looked at my indoor cultivated palm tree. So he always advised me, why don't you look at the roots of this? And I told, well, we can, but what can we do with palms? We cannot grow them in our country and so on. But then I did. And I was really surprised. And it was then connected with this collaboration in Saudi Arabia with palms, different species. And we found some interesting things in one of these species, which was presented during one of the silica conferences, because it is also something which is interested for a special group of people dealing with silica and plants. So then we put a big group of people together to study also molecular biology and several aspects, as you can see here, and we focused on the date palm, very important species in dry regions. And this was really something which we didn't expect. And what is here as a specialty is concentration of silica in very specific cells which are joined with sclerenchyma. And this is also still not very clear what is the role. But if you look at these palms and you see the stability of it, so it may be related somehow with this. But there are still many questions. And now I will ask probably Michal to continue, because if I remember well, we have had a few 3 slides in connection with this silica story.
Okay, thank you very much for this opportunity. Yeah, when I met first time Phil, because this meeting is about him, I was a young, very stupid student. So it was like 2002 or something like that, and so I was knowing him for over than 20 years, and our former professor of our department asked me after his lecture, please go with Phil somewhere outside. So I took a friend of mine, and okay, probably the professor meant that we should go for a beer with him, but we went up to the mountains, and so we took him crossing the forest and so on, and went up to the hill. There was nobody. Phil was totally muddy and probably a little bit exhausted. And later on at the evening, he told me, you know, I like walking, but only from the kitchen to the probably other rooms inside the house, but not really passing several kilometers in the forest. So yeah. So at the time it started, and yeah, his knowledge was very helpful on the background of this ionon characteristics and importance of silicones for plants, and we established several experiments, and we were testing, for example, pathogenic fungi attacking some cereals, and we found out that it is possible to improve the health of a plant by adding a silicone, and to improve the fitness of plant, and after that the plant is able to withstand this attack of fungi. And later on we continued to check where is the boundary where to stop the fungi, and we found out that it was this exodermal layer if the silicone has been present. Otherwise the fungi was able to penetrate even more deeper. And nowadays we are continuing not with pathogens but with pests, and this is something else, but we see something similar, that to improve the health of the plant can help and save the life of a plant after a very massive attack with pests. And we were cultivating those plants like just 4 weeks, and we were getting from one single aphid from 500 till 1000 on each one plant. So and the plants having this silica inside, they were able to withstand this massive attack. So it was very nice, and it is on behalf of Phil ideas, and of course of Professor Lutz as well. So
yeah. So well, this picture was taken to the publication by Martin, and it is one of the unfinished discussion which we had in my greenhouse in front of my collection of plants of these cacti, because we discussed what was in the previous presentation. So we have to continue and remember him, and this is really the absolutely last publication which we had together with him, the favorite calcium. So thank you very much for the patience and our missing the right time. We were very happy that we could run this like this. Thank you.
Thank you very much, Alex. Should we, no more questions online? Was there one question from the room at all? Ken? Oh, was there one online?
Rob Brooker has it.
Ah. Rob, do you want to unmute and ask?
No, it wasn't a question actually, thanks.
Okay, fair enough. So if we've not got any questions from the room, I suggest we go for lunch.
We'll move on to the main event of the day, which is the Dundee Root Medal Lecture, which will be given today by Professor John Hammond from the University of Reading, who was a PhD student of Philip's in the day. So I think he can relate to everything that's been said today already. And John's going to give us a bit of a retrospective on Philip's work, but also how it's influenced his own work, I think, as well. So, John, we look forward to hearing what you've got to say for the next 45 minutes or so, and then we'll have plenty of time for questions. So, thanks, John.
Thank you. So thanks, Tim, for the introduction, and good afternoon, everybody, and welcome to everybody who's joining us online. I think it goes without saying that many of us would rather be here with Philip taking the stage today and giving his retrospective on his career himself, rather than me standing here. However, it's a pleasure and an honour to be here and in a position to come today and do that. And that's a position I wouldn't be in if it hadn't been for Philip's mentorship and support and friendship over the last 20 years. So I hope I give a talk that Philip would have been proud of, and not one that would have drawn one of his famous stares. We've already heard a great deal about the legacy that Philip left behind through the talks over the day, and I hope now to sort of piece that together and draw some of those dots together and look at how that career progressed, and those thought processes that we've been talking about, and his ability to conceptualize hugely complex areas of science into very understandable concepts developed. So I've broken down the talk into a few sections. First, I want to give some context about why I'm stood here and how I came into Philip's life. I look back at Philip's prolific publication history. Those of you that knew Philip will have known his obsession with citations and metrics, and what was getting cited and what wasn't getting cited more often than not. And then some areas of research which I think summarise the sort of big chunks of Philip's career, that working on cations and membranes in his early years, and then work around the sort of uptake of nutrients into and the sort of phylogeny work that Conrad was talking about, and then the work that I worked on with Philip from my PhD onwards around phosphorus. So first, some context. So this was the late 90s. I was an undergraduate at the University of York. I was being taught by Ottilie Liza, Dale Sanders, and Simon McQueen Mason, and this was the sort of on the precipice of the genomics era, and this little fellow was making a big splash in the plant science community. I'm obviously talking about Arabidopsis, not Philip, although I'm sure he was making a splash in his own way. So Arabidopsis featured quite heavily in my undergraduate teaching, and I was, encouraged and inspired to go into science and wanted a PhD. And I thought, well, if this is the next big thing, this is something I should be focusing on. And Philip had a PhD opportunity advertised, which I applied for, called Smart Plants and Phosphate Nutrition. And it was an eclectic mix of supervisory team. So I had a plant physiologist in Philip, an ecophysiologist in Martin, a plant molecular biologist in Malcolm, and Clive in the bottom right corner there was a horticultural agronomist. So very diverse mix, but Philip was certainly front and center in that, in leading that team, and hearing people talk about those Friday afternoon conversations and spending hours and hours in Philip's office with his one finger typing, constructing manuscripts and sort of trying to predict what he was about to say or about to write on the page, and thinking, getting it wrong every time, and he'd come up with something else. And so that's where it started. This was 2000. I joined Philip's group at Wellesbourne, and I started my PhD. And we'll come back to that at the end of the talk. So Philip's publication history. He was a citation classic in himself, and as it's already been referred to, he was one of the named in the highly cited list of researchers from 2014 to 2019 inclusively, a fact that he was immensely proud of. And as it says on their website, once you're listed as a highly cited researcher, you're always a highly cited researcher. Philip's research covered 5 decades and over 300 publications to his name. I've struggled to find a definitive number. All the databases have slightly different numbers, and you'll see the citations vary from 30,000 to 45,000, depending on the database you're looking at. And so this has been a hugely daunting task, is how to synthesize 5 decades of research and 300 publications plus into a 45-minute talk about his career. And I'm sure I will have probably picked some papers that, or omitted papers that people think should have been here, but I hope they've been covered, certainly in the talks that we've seen earlier today. So yes, regularly publishing double-digit numbers of papers annually, and 3,000 plus citations a year. He was a prolific writer in that sense. And it wasn't a sole operation. Many of his publications, especially early on, were sole author papers. But Philip had co-authorship with over 600 academics globally. This is a network diagram of those interactions and linkages between his co-authors. And hopefully you'll see some familiar names up there, and certainly some that have already spoken today. And if you delve into this, you can start to see networks evolving around institutions that he worked and collaborated with, but also areas of research that he influenced and was sort of instrumental in developing. And I think it would be fair to say that influence was global, not in just who he collaborated with, but in terms of who cited him and his work. So I've got 28,000 citations here from Scopus. But yeah, if you look at, or Web of Science, but if you look at Google Scholar, it's 45,000 plus. But his research was cited from the southern tip of Argentina, Rio Gallegos, to the Norwegian archipelago of Svalbard in the north. And if we just zoom in to Europe as an example, every country in Europe, there is someone who cited a piece of Philip's work. So we've heard a snapshot of what Philip has been involved in over his career, and it took him into many avenues of research. And this is taken from Martin's In Memoriam piece that was published in Plant and Soil, a word cloud of the titles of Philip's papers. And fittingly, root is front and center for this diagram, but you can also see a number of elements and plant species and aspects of the work that he and the tools that he used in his work. So for the focus of this, the title of the talk is Elements of His Life, and elemental nutrition of plants was his core research area, and something that didn't waver throughout the course of his career. And so I've just highlighted in the diagram the elements that feature most prominently in the titles of his publications. But this isn't an exhaustive list. I think when you look in detail at all the 300 plus publications, I found references in the titles to all the macro and micro elements of plants, plus cesium, silicon, iodine, cadmium. So given Philip's work ethic and prolific publications, as I said, forgive me if I don't cover every part of Philip's research over the next half an hour. But and I think if I was probably just to stand here and read out all the titles of Philip's papers, I'd run out of time before we got to the end of the session. So, as is customary, I'm going to start from the beginning, or near the beginning. And this is a cover from a plant journal paper that contained a paper by that Philip was involved in, and I remember this being stuck on the wall in Philip's office at Wellesbourne. He's immensely proud of that. And this was in the days of black and white, or if you wanted a colour image in a paper, it was going to cost you. So one of the easier ways to do that was to sell it to the journal that you could get it on the cover. And there was often either no charge or a lesser charge in that. So Philip's early work focused largely on the uptake, translocation and compartmentalization of mono and divalent cations, particularly potassium, calcium, and magnesium. And this diagram's taken from one of his earlier papers. Apologies for the poor quality. I couldn't get a good PDF of it, unfortunately. Looking at the effect of root temperature on the and root to shoot ratio on the uptake of potassium in the roots of rye. And this is from his PhD work that he did with Mike Earnshaw at Manchester and David Clarkson at Long Ashton as his supervisors. And I think it reflects quite nicely on some of the work, the presentation Tim gave earlier on his ability to communicate and simplify and conceptualize a lot of this work. And that frequently took the form of either mathematical or mechanistic models that would allow you to sort of draw together this sort of complex mix of data to get a logical conclusion out of it. So the potassium carrier is depicted here, influenced by the temperature of the root, but also then the shoot to root ratio, which alters the essentially the sink source relationship in the xylem and via the concentrations in the root, and the carrier number influences the carrier activity to also influence the uptake of potassium. So as Philip's work progressed, it focused more and more on those underlying mechanisms controlling the uptake and compartmentalization and efflux of these cations from the roots. But he always kept with him that whole physiology perspective and didn't get lost necessarily in the mechanics of the individual transporters and channels that he worked on. So here's a paper from 1990, jointly co-authored with his future wife, Jackie. And this was with Andrew Smith whilst he was at Edinburgh during his first postdoc, where they focused on the magnesium dependence of an inorganic pyrophosphatase proton pump and its likely role as an ancillary proton pump to the proton ATPase. And the hypothesis was that if the ATPase driven proton pump ran out of substrate, the magnesium could be moved through this inorganic pyrophosphate proton pump. And so as he progressed, this is a slide from one of Philip's earlier lectures that I think he gave to me when I was at Wellesbourne. looking at the electrophysiology techniques that he had been exposed to in Edith McRobbie's group when he was at Cambridge, and started to utilize these cutting edge technologies to now drill even further down. So we've from roots to proton pumps to now understanding the properties of individual channels using patch clamps, planar lipid bilayers, and the early versions of sort of cell sorting capabilities based on their expression are in different cell lines of GFP. So using these approaches, Philip began to identify and characterize electrophysiologically the channels that mediate the transfer of cations across the plasma membrane, and understand the direction of flow and the pharmacological properties that inhibit or allow these processes to occur. And certainly a lot of my early teaching in Philip's was trying to understand these voltage current diagrams, and I'm not fully sure I still fully understand them, but it was a way, it allowed them to, Philip to understand what was moving across the membrane and how. And so this is one of these first papers that he published using this technique with Mark Tester, looking at the properties of individual channels. And these, again, were in the roots of rye plants that had been incorporated into planar lipid bilayers. And so when the channel is in one of these planar lipid bilayers, you can manipulate the current flow and measure that current flow using electrodes either side of the membrane. And this first figure on the left shows the several different channels and their properties that had been incorporated into one of these membranes. And this is in a pre-genomics era, so we don't know, this hasn't got a gene, it hasn't got a name. They were just characterized by their actual capacitance. And so the figures on the right-hand side show one of those channels, P49S, which had particular interesting properties in terms of its ability to transport cations. And in this paper, they showed that it was able to transport potassium, and that it was inhibited by quinine in a voltage-dependent manner. So taking advantage and coincidental with his move to Wellesbourne and the interactions with colleagues such as Malcolm Bennett and Dale Sanders, he was very keen to utilize the technology and the modern molecular approaches which were being developed in Arabidopsis to be able to understand in more detail. So previously the channels were being extracted from root cells, and it was very difficult in terms of understanding where that channel belonged in the root system. Was it an epidermal cell? Was it cortex? And the processes that channel might be functioning in terms of transporting cations across the actual root. And so this paper took advantage of the green fluorescent protein, which was being utilized to mark cells and look at the expression of genes. And in the diagram here, on the black and white diagram, again, free color prints in journals. So this is the same, this is from the Plant Journal paper, which had that cover image at the beginning. You can see the stellar cells in the cortex are marked with the GFP, allowing the selection of different cell types and then the cation transport functions in those different cell types to be characterized at even greater depth. And so this GFP allowed that selection of those plants, showing the different cation transport properties of these non-fluorescent cells versus the fluorescent stellar root cells, and the different inward and outward rectifying potassium transport that was occurring in these different cell types. So much of Philip's work during this period focused on voltage insensitive cation channels, and writing a commentary piece in 1999 on their potential roles in regulating sodium influxes into plants and their inhibition by calcium, and the potential for those to be compensatory fluxes that allowed osmotic adjustment or independent, which were independent of the membrane voltage. He later went on in collaboration with Romola Davenport at Cambridge to show that these channels were actually permeable to calcium under physiological relevant conditions. And through barrier modeling, which is depicted here, and one of where the energy of the transport of the cation through the channel was modeled, and through planar lipid bilayers, to show that the this potassium channel was contributing to the influx of calcium into the cells. And I think it would be remiss of me not to mention Philip's paper from Philip and Martin's paper from 2003, where he synthesized a lot of this knowledge about calcium transporters in plants. And certainly one of his highest cited papers is attracting 2,000 plus, I think 2,600 citations to date on that. So, you know, and that was his ability to summarize and extract that knowledge in a way that I think very few of us can truly do in a literature review. And Philip's literature reviews weren't literature reviews as we've seen already today. He always wanted to take them to the next level and bring in that data and knowledge that only he could do, and present it in a way that hadn't been presented before, to challenge the dogmas and to drive the research forward. So just coming back then from that very detailed molecular electrophysiology work to the whole plant physiology again, before I finish on cations and membranes. This is from a paper published with Lim Ho in 2005 on the cellular hypothesis of blossom end rot in tomatoes. And Philip's knowledge of calcium transport and movement, it was able to illustrate that this phenomenon in tomatoes was directly linked to that inability of calcium to move freely through the phloem, because it's a living tissue, and that interferes strongly with the calcium signaling processes in that. And so that whole process, that lack of supply to the growing tissues, reduces the apoplastic calcium, which in turn reduces the loss of membrane integrity, leading to solute leakage and eventually cell death. And again, it's another example of one of Philip's sort of conceptual models that he was very keen to draw out, and that we've seen several times today. So this brought us up to the early 2000s, and these two handsome chaps turned up at Philip's door. And I think this is where we sort of Philip's research shifted a gear and went down a different avenue. And that was following a talk that Martin gave at Wellesbourne, newly arrived at Wellesbourne from his PhD looking at cesium accumulation in plants, and the evolutionary controls that may be in place on the accumulation of those elements, and how you could use that potentially, that choice, that knowledge about the phylogeny, to choose plants that may be more adapted or better suited to phytoremediation, or enhancing the elemental composition of nutrients in the edible portions of the plants to improve their human and animal dietary intake. So I'll start with the Martin and Philip's first research paper. They published a couple of reviews before this on cesium uptake and chlorine, but this is one of their first sort of attacks at the literature in the development of this phylogenetic theory around the influence the phylogeny has on the uptake of nutrients. And this is focused on the heavy metals and identifying the fact that a high proportion of the variation in, for example, nickel and zinc occur at the order level or above. And also the observation that several of the orders contain more metal hyperaccumulators than others, in particular the brassicales. Through Philip and Martin's discussions, a plan was hatched to test this further. And then Conrad's shared a similar picture earlier, but this was the original phylogeny experiment at Wellesbourne from, I think, 2001. So I'd only been in my PhD for 12 months, and I was roped into helping out with another crazy experiment where we were looking to grow 144 species representative of the angiosperm phylogeny, and the challenge of sourcing the seeds, working out their germination strategies, and putting some in the freezer, some in the heat, some of them needed smoke, just to get them to germinate and all be growing at the same time in this NFT system. But this was complemented by a Herculean literature study covering 244 studies and 61 papers in what we've seen as already one of Philip's unique abilities to sort of, keep all of that knowledge in his head and be able to relate these papers and concepts across such a diverse set of literature. So we grew a huge selection of plants, and from cacti to crops to plants that are accustomed to growing in deserts. And what we showed was that there is this inherent effect of the phylogeny on the accumulation of elements within it. This data is looking at the experimental calcium content on the bottom axis, and on the y-axis the data from the literature, showing good correlation between those. But at the order level, or the informal group, sorry, you can see that separation already between eudicots, commonoloid and non-commonoloid monocots in terms of their calcium content. So you can start to hypothesize that if you wanted to start feeding the population more calcium in their diets, using asparagus is probably not the best strategy, but looking at maybe some leafy vegetables might be. And so again, the brassicales came out in this paper as being an interesting group of plants to look at. It had one of the highest calcium content in plants. And so that started a whole area of research looking at how you might enhance the calcium, magnesium, other nutrients into the diets of the human population. And brassicales, particularly interesting because, as we've seen already, calcium doesn't move through the phloem. So trying to put calcium into the seeds or fruit of plants can be quite challenging. But as it moves freely in the xylem, it moves quite nicely into the leaves of brassicas, and one of the vegetable brassicas are typically leafy vegetables that have the potential to accumulate more calcium in them than other plants. And so, given the proximity of Wellesbourne to the national or the global centre for genetic resources in small vegetable crops, we had at our disposal large collection of brassica plants, brassica genotypes, sorry, from the vegetable brassicas. And that allowed us to start working our way into the brassicales in a similar way that Conrad has done with the caryophyllales, to look for opportunities to increasing that calcium concentration in the leaves and of plants. And so we did that across a diversity foundation set, which represented the vegetable brassicas that were in the gene bank, and also a mapping population. So we were able to start identifying genomic regions that were associated with these traits. And these are the different subtaxa of the brassicas that we were looking at, and their variation in leaf magnesium and leaf calcium within that. And from that, we were able to identify again that calcium and magnesium were particularly heritable traits. They had a strong genotypic variation associated with them in comparison to some of the other elements that we were looking at. So there was potential to breed for that and develop strategies to increase the calcium and magnesium in the leaves of these plants. So I'm going to take you down one avenue of that, and that was, I think, spurred on by Philip's knowledge of this protein membrane transporter in the vacuole, the CAX protein that loads calcium into the vacuole and keeps it away from the cytoplasm, where it can interfere with those cellular calcium signaling processes. And so this is the CAX1 protein from Brassica rapa, showing its transmembrane structure, and the N-terminus, which is an auto-inhibitory domain that had previously been shown to disrupting this can cause the accumulation of calcium into the leaves or tissues where it's functioning. And keeping with the times and moving genomic technologies and genetic resources, the tilling pipeline had become available in Brassica rapa that we were able to use to target and identify mutants in this this protein. And so the dots in red are lines where we managed to identify a mutation in this protein. And that allowed us to then identify lines which may or may not, as a consequence of that mutation, increase the concentration of calcium in the leaf tissue. And so one of these lines, line 12, which had one of the mutations in the auto-inhibitory domain, as had been shown in Arabidopsis, did in fact accumulate higher concentrations of calcium in that material. Concurrently with that work, that was a reverse screen. We knew the target, and again, one of these crazy ideas that I think probably Martin and Philip cooked up in the pub was that, well, we're growing these material anyway, so we'll just screen all of them for all the nutrients, which sounds trivial, but it was a massive undertaking of logistics and growing and statistical design to make sure that they were replicated appropriately. But what turned out from that screen was the identification of several mutants or several mutants in that tilling population, which actually had altered phenotypes for other elements, in particular magnesium. And so through a long and winding road of mapping and backcrossing and double checking and resequencing, we were able to, through that process, identify the underlying mutations that were giving rise to these higher calcium and higher magnesium concentrations in this SGN3 line here, higher magnesium, higher calcium, as a consequence of this mutation, which turned out to be a gene called SENGEN3, which is really critical for enabling the Casparian strip in the roots to fuse into a single continuous band. And the consequences of that are that that barrier that is normally in the endodermis controlling the flow of nutrients into the plant is disrupted and remains open for longer or further up the root, allowing more calcium and magnesium to move into the plant tissue. So as well as the sort of genomic and genetic approach, Philip also worked quite heavily on the agronomic side of things as well. This is some data from a trial that I think we did at Wellesbourne, but it might have been subsequently repeated elsewhere, looking at selenium uptake in wheat, and the more you put on, the higher the selenium concentration you get in the seeds. And then following his move to Dundee, focusing more on potatoes, looking at the application of foliar zinc fertilizers to increase the zinc concentration in the tubers of crops. So that brings me back to the title of this section, Eats, Roots and Leaves, which was the title of a paper Philip and Martin published in Proceedings of Nutrition Society around the potential for these approaches. And this was really sort of that, yes, we can do it, but what are the implications of it? And these figures show the calcium, magnesium, potassium intake, dietary intakes for males and females in the UK population. And what you probably can't see on these diagrams is this, the dotted lines are the increase in that nutrient you get from adding additional portions of fruit and vegetables into your diets. And the dashed black lines are when you biofortify those portions. And you can see what's critical here is the is shifting the population from this below this lower reference nutrient intake, and by increasing both the proportions of fruit and vegetables or by biofortifying them, in many cases you move most of the population out of that lower reference nutrient intake. So back to these two chaps. My turn now. And as I said, I joined Philip's group in 2000 as a fresh-faced PhD student, and got the shock of my life in terms of what I'd taken on with Philip. It was certainly a daunting experience as a fresh from my undergraduates to be challenged in the way that Philip challenged me, and to have those conversations with him and not never go in a direction that I thought they would go in. But my project was entitled Smart Plants and Phosphate Nutrition, and I guess this is a rebalance of Philip's membrane potential in that from cations now to anions. And prior to this, Philip had not worked on anions or phosphate, so this was a new avenue of research that began with this project. And so a smart plant was the concept that you could use the plant to inform you of its physiological status by driving a marker gene, such as GFP or GUS, with a nutrient or stress responsive promoter, so that when the plant became stressed, it activated that promoter, and you would be able to detect the GFP or the GUS in the leaves of that plant, and understand then the physiological status of the plant, hopefully prior to it showing physiological damage to the plant. So plants have evolved a number of different mechanisms for coping with low phosphorus availability in the soil. And this is those summarized in a paper Philip wrote most recently on improving phosphorus utilization efficiency in plants. And one of those areas is actually how it manipulates the pools of phosphorus in the tissues, in the leaf tissues. And a couple of years prior to me starting at Wellesbourne, Christoph Benning's group had shown that when plants are phosphate stressed, they start to swap out phospholipids in their thylakoid membranes for sulfur and galactolipids. So, and they published this paper that showed that one of the key genes in that biosynthetic pathway, SQD1, has a higher expression under low phosphate conditions. And this is the key rate limiting step in converting in the production of sulfolipids by incorporating sulfate into UDP glucose. to produce UDP sulfoquinone. And so just before I arrived, master's student Kath had cloned the promoter from this gene into a vector that was driving GUS or GFP. And so my PhD was set to test this out in Arabidopsis. So I would just naively thought I was just going to go and grow some plants and measure some GFP, but Philip was very keen to make sure I didn't lose sight of the plant physiology and didn't get wrapped up in the molecular biology of these things. And so we spent a long time optimizing the assay conditions and making sure that we knew what the plant was doing when we were testing it, and optimizing the growing conditions for Arabidopsis. And what we showed was, well, what we published in my first author paper back in 2003 was that when you take phosphate away from plants, the GUS expression increases as we would have predicted. And so there's a concept. This was there, and it was a tested hypothesis that we completed. That relied on a single gene and a single promoter, and it obviously had to be transformed. And at the time this was the early days of the genomic era, and microarrays were coming onto the scene as a way of not just measuring single genes, but measuring the whole complement of genes in their expression. So building on that work and through sort of detailed conversations with Philip, the concept of monitoring phosphorus stress through one gene through a reporter, the next logical step was to then develop a whole pipeline through microarrays to test to monitor the phosphorus stress of a plant. And so we did this in potatoes, again looking at the transcriptional profile. This is some the sulfolipid and galactolipid pathway showing increased expression of SQD1 during under phosphate stress. And through this data that we collected, Philip was very keen to be able to use that data in a way that would predict the status of the plant's phosphorus status. And so we used a machine learning approach we were able to take that data and use that data from not just one gene, but 30,000 genes to predict the status of the plant. So these were potato plants grown in the field, either fertilized or unfertilized. And the values here, or the support vector machine margin values, indicate whether the plant was P deficient or P sufficient based on the expression profile of the plant. And this is matched with the lead phosphate concentrations presented below. So we were able to do that, and the reviewers came back and said, well, that's nice, but what about all other stresses? What happens if this doesn't work? And so I was quite stressed. I was thinking, well, how are we going to do that? We can't grow the potatoes again. And we've got this. So Philip said, well, there's all this data in Arabidopsis that's now freely available online. Can you test it with that? So we converted the diagnostic genes that we developed into their Arabidopsis homologs, and we're able to show in the Arabidopsis data set that when there was data from low phosphate stressed Arabidopsis, that again, the model that we developed was able to predict the status of the plant based on the gene expression. So coming back to brassicas, as well as doing a lot of that sort of molecular work and the work with the microarrays, we're very interested in the phosphorus use efficiency and the traits that underlay that within brassicas. So using those genetic resources that we use with the calcium and magnesium, applying that to phosphorus. And so that work really ran parallel to the calcium and magnesium work, and that we were able to screen diversity foundation sets, commercial cultivars and mapping populations within that for different traits. And this data is an example from agronomic phosphorus use efficiency in those systems. And during that time, we took on a visiting postdoc from China, Professor Shli, and that spurned a sort of a huge collaboration with Philip and China. And Philip really enjoyed going back to China and talking and giving lectures at Hwazong Agricultural University. But that work started in Wellesbourne on brassica roots, and these are some data showing some of the early filter paper screens that we were doing with the brassica napus lines, in this case of Tapidora and Ningu, looking at the differences in root architecture between those parents and their responses to phosphate availability in that. And then most recently, that work led on to probably highlight one of the more recent publications with Schlee's group and Philip and myself, testing some of the hypotheses around local and systemic signaling in plants in response to low phosphorus availability using split root systems, something that Philip and I had talked about during my PhD, but never got the opportunity to fully test it. And so this was published last year, and it allowed us to look at and identify the genes that were upregulated and downregulated locally and systemically within the root tissues of these plants, but really look at how those processes were communicated from the parts of the root system which were phosphate stressed versus the ones that were phosphate replete. And it provided an opportunity for us to go back to a paper we published in 2006, looking at the biomass accumulation of under different phosphate stresses, and showing that actually under nitrate and phosphate stress, the root to shoot ratio increases markedly compared to what happens under potassium stress. And we hypothesized and published a couple of review papers on the role that sucrose and carbohydrates play in driving that biomass allocation in the roots in response to these stresses, both as a carbon source for the increased biomass that we see, but also as a potential signaling molecule in that. And so within this split roots study, we were able to apply that sucrose, the low phosphate compartment of the split root system, and show that actually in the phosphate replete side of it, it actually increased the total length and number of secondary lateral roots that were being produced in that compartment, signaling to the plant that that's to mine more of that phosphorus from the soil. And I think I've alluded to it already, but Philip's ability, and Tim did as well earlier, to sort of conceptualize and simplify many complex processes. And this was one paper I wanted to highlight that Philip wrote with Hans Lambers and Jinbao Shen last year, looking at root exudation in below ground economic traits for resource acquisition in plants. And I was really struck by this diagram that was at the end of the review, and how incredibly sort of simplified they'd managed to make what is a huge data set and knowledge in terms of the strategies plants may use to acquire nutrients from the soil, and that's those plants that were able to do it themselves through exudation of nutrients versus those that outsource that to other soil microorganisms such as mycorrhizal fungi and bacteria, and how those processes are delineated again at with those phylogenetic evolutionary history traits that were present long before as these plants and groups of plants evolved to cope with their environments. So I just want to finish off by highlighting one that sticks out as a sort of an anomaly in probably Philip's publication history, and a piece of work that Philip and I did when we were at Wellsbourne. And just for the enjoyment that I had and seeing Philip's ability to switch from sort of molecular biology processes to landscape-based transport processes using very similar process models. And so this was a piece of work that we were commissioned to do by DEFRA to quantify how much phosphorus goes into the rivers and surface waters of the UK. And it was very contrasting to all the work that I'd done with Philip before. And it really stuck with me as a very sort of complete piece of work that we were able to achieve using very simple mathematical processes to calculate how much phosphorus goes into the rivers in the UK from different sectors. So agriculture, household, which is sewage, industrial and background sources. And we did that across the UK's what were river basin districts at the time in preparation for the EU Water Framework Directive that was coming into force. And what DEFRA wanted was a very simplified mechanistic process that allowed them to tease apart the numbers in a way that they weren't able to do with some of the models that were currently being developed for this. So this was an Excel spreadsheet that we used to calculate these sources of phosphorus and where they went. And this just shows the large dominance of sewage treatment sources that went into contributing to phosphorus that went into the rivers and lakes of the UK. And that's very strongly correlated here. This is total phosphorus and soluble reactive phosphorus with the area normalized population density, sorry, of that. So more people, more sewage, more phosphorus going into the waters. Now, I have to say that that's certainly shifted a little bit in recent years in terms of some of the processes that sewage plants use to to capture some of the phosphorus, but it's still a pertinent issue with the amounts of sewage being leaked into rivers that we've seen quite strongly in the press recently. So I wanted to finish on that as a little anomaly to the rest of sort of Philip's publications, and leave it there and thank you, as Philip always did, thank you for listening. It would have filled the screen if I tried to list all of Philip's co-authors and thank them individually on there. So if you don't see your name, I can assure you your name is in that network somewhere. So thank you for listening.
Thanks, John. That was a great job of summarising a huge body of work, and I think we've seen from that the breadth, but also work from molecular all the way through to landscape, from fundamental through to applied. I mean, everything's there, right?
Yeah.
So we have time for some questions for John. Are there any questions online at all? No. And I mean, put your hands up if you wish to make a question online, but otherwise in the room, any questions? Or is everyone just completely overwhelmed? There's one online, okay. Jambo. So Jambo, it must be very late with you.
Yes. Thank you so much. Professor John Hammond gave a very nice presentation.
Thank you.
I would like to take this opportunity to give a big thanks to Philip White Philips make a great contribution to the collaboration with China Agricultural University, especially on phosphorus story. We have a very good collaboration with Philip White. So Philip visited us several times and then gave many lectures and then have a very intensive discussion with our group and also with our colleagues. All our colleagues have a good memory with Philip. So we also have several publications with Philip. I think is a really pity we you know Philip passed away last year. So I really show my deep feeling, give a big thanks to Philips, and thank you for John to mention our publication with Philip. Thank you.
Thank you. I know Philip really spoke quite well of his time in China and his visits, and he really enjoyed those conversations and developing and writing those papers with you.
Yeah, Philip is very happy. Have a very good time in China. We have a lot of discussion and then talk science and even culture, custom, and a lot of story. Thank you so much.
Thanks, Jennifer. So there's a question online, I believe.
What's the title of each book? So the panel on the book stressing the importance of country.
Leaves. Yes, it was.
Right, fitting.
Yes. And important not to keep the comma in there, I think.
Yes, exactly.
It gives it a different meaning altogether.
Any other questions in the room or online? Glenn.
Just wondering what's your opinion in the light of your own work and Philip's work of the years, should the next priorities be? It's been easy.
Yeah, I think that's whatever I say. I think probably Phil would come up with the antithesis of it and say, no, you're wrong. It's this. But I think that's yeah. I'm going to dodge that one.
I think one of the things that's obvious from today is that there's so many different avenues that we've worked on that it could be anything. You know, it could. I mean, we're all probably going to take a different route, and it all stems from our interaction. Oh, a lot of it stems from our interactions with Philip. So lots of different directions. But I mean, that's a.
I suppose okay, another maybe a slightly different question is what do you think we as scientists ought to learn from Philip's approach?
I think certainly what Tim said in challenging the dogma, and certainly that ability to sort of, you know, Philip was always keen to when he was doing his reviews, it wasn't just the last 5 years of what's happened. It went back. Jackie was telling me at lunch that she's got piles and piles of manuscripts that Philip had poured over to do this. And I think that's certainly not losing sight of what has gone before and building on that in a way that I think some reviews don't do that justice. I think one thing that I was reflecting on looking through his papers was that sort of switch from the electrophysiology and then the molecular biology sort of took over, and you could sort of possibly infer through knockouts or overexpression that what the properties of some of these transporters and channels were. And I think that sort of there's an opportunity to go full circle back to sort of to look again. Now we know that and spend more time on that electrophysiology element of some of these transporters. I think that's certainly something that's not been revisited as well as it could have been.
I mean, I assume everyone in the room would who's worked with Philip would pick out the fact that his attention to detail was unbelievable. Right? I mean, the most. I mean, it was just
so demoralising.
Yes,
When you just you think you've done your best piece, and then it comes back and it's just covered in red.
I learned from Philip that in Australia we don't speak English.
That's true. We all know that.
Relearn my mother tongue.
Yeah. Okay, so if there's no more questions, then we'll move on to the ceremony part of this, the medal handover, and Martin's going to come up and give us a few words. Maybe we can turn this around so people online can see what's happening. So Martin's going to say a few words and hand the medal over to John on for the.
Thank you.
Martin.
Can we try and recreate that image? Back the doors.
Leaning in.
Should we face into the camera then? Yeah, well, thank you, John. I thought that was, a fantastic overview. I'd forgotten so many parts of that, and I thought you brought that back to life incredibly well. Just going back to Philip, I was wondering, my mind was sort of whirring at the back there, thinking about some of the latest sort of advances in AI and natural language processing, and maybe technology will be able to sort of give us tools to be able to kind of start to work on, if we can put those 300 papers into a
chat GTP and that's get to
so I think that those types of approaches will obviously emerge as we go along. I think I've probably said things that I wanted to say in the memoriam piece. So I don't think, and I think everybody here today has said the things that were really important to them, and I think it's been a fantastic set of talks. So thank you for that. So I'm going to, on behalf of Philip, hope you can accept. It's the 9th Dundee Route Medal Award on behalf of the Society, and it's a great privilege to be passing this on to you.
Thank you.
You might want to open it up so people quality.
Oh.
So we're excited to see it. I mean, people online.
But it fell out then. It's all it's the camera. There we go. Those online.
There we go. Okay, so thanks, Martin.
Thank you.
Thanks, John. Thanks, everyone, for coming. It's been a really interesting day and a worthy tribute to Philip. So thank you all, and I hope you have a safe journey home. Thanks.